ये वाला लूं? अरे
वो ऑफलाइन प्रेजेंट करेंगे, उनका नाम लिस्ट
ये है मेरे पास।
उसे सबसे देखना होगा ना कि पीपीटी सबका रेडी है।
एक मेरा अच्छा सा
सारा
ये ले लो। इसमें अच्छे से नहीं आया है, वो तो नहीं आया, बस ये तो आया है ना। एक कौन सा है, मैं अभी कर लेती हूँ कि कितने प्रेजेंट।
फोन नंबर का मतलब कि से बात कैसे पूछते हैं फिर।
I'll be reading out their name, those who have been shortlisted for presenting their paper online, offline, here on the auditorium on stage. So please do respond for your presence. Deepak Mishra, Deepak Mishra, Vaishnavi, Vaishnavi Garg, Riddhi Manu, Vaishali Kausal, Vaishali, Samir Beg, Vaishali Kausal. Do we have Vaishali Kausal over here? Samir Beg, Sahbat Alam, Sahbat Alam, Karuna Shankar Rathore, Karuna Shankar Rathore, Dheeraj, Dheeraj, do we have Dheeraj? Subhangi, Subhangi Jarawat, Shivanshi Chauhan, Shivanshi Chauhan, Zoha Siddiqui, Zoha Siddiqui, Monica Moitra, Archit Akhil. So we have got one attendee with us. Others, I would request you to please mark present Archit Akhil. Until now, we have only one presenter available with us out of the 15 presenters. सेकंड आ रही है तो आएगा, दीपक आएगा, मोइत्रा भी आएगी, प्रीति आ गई ना, तो आएगी सर, वो लोग आएंगे, इंटीग्रल के ही है, मस्ती ठीक है। ये ऑफलाइन वाले आपने मुझे नहीं भेजा था ना, ऑफलाइन नहीं भेजा था क्योंकि ये दे दिया मैंने, आदर्श को तेज निकाल के दिया, आदर्श लग गया यहाँ पे ऑफ लग गया। All the attendees here in the offline mode are requested to move for breakfast. It has been arranged for you in the back of the auditorium as usual, like yesterday. I request Everyone over there, please be settled down. We are going to begin with our event shortly. I will request everyone to please be settled down. Student volunteers in the discipline committee, please take care of the decorum of the hall. Start करें मैम start करें. A warm good morning to you all. Hi, Dr. Lucy Mahapatra, Associate Professor, AIP, AUUP Lucknow Campus. Welcome you all on behalf of Professor Dr. Anuradha Mishra Ma'am, Director, AIP, AUUP Lucknow Campus, and the organizing 40 members of this 2 days national conference on recent breakthrough in pharmaceutical sciences from bench innovations to laboratory applications and beyond, either RBPS 2025, organized by MIT Institute of Pharmacy, MIT University, Uttar Pradesh, Lucknow campus on 24th and 25th February 2025. Thank you to each and every one of you for joining us both online and offline today, as we are truly honored by your presence. May I now take this opportunity to brief you all about the various endeavors that we are going to undertake today, both at the online Zoom platform and offline here at the auditorium of AUUPL. To begin with, we will get started with all our technical sessions by our revered and expertised speakers in the following sequence. The first technical session of Day 2 RBPS 2025 includes the talk of Professor Ajay Sharma, Professor and Head, Department of Pharmacology, DPSRU, New Delhi, to be held at the auditorium. Following that, we have the second technical session, including the talk of one of the most revered speakers from Jamia Hamdard, Delhi, Professor Dr. Vibhu Prasad Panda, Professor Pharmacology and Phytochemistry, SPUR, Jamia Hamdard, New Delhi, in the auditorium again. However, Dr. Deepak Chitkara, Associate Professor, Department of Pharmacy, BITS Pilani, will be joining us online on the Zoom platform for the third technical session of the day, RBPS 2025. In the post-lunch hours, we will have the offline oral presentation of our registered and selected participants in the auditorium in between 2 to 4 p.m. Following that, we will be ending this day, 2-day extended national conference with its valedictory function to be held at 4 p.m. in the auditorium, wherein all the winners of oral and poster presentation competitions will be awarded with the cash prizes on stage. Respected dignitaries and dear delegates, it might be known to you all that speakers at a conference are crucial for the reason that They are the primary source of information and engagement of attendees, delivering expert insights, sharing their new knowledge, and inspiring the audience with their presentations, essentially driving the overall success of the event by providing valuable content and creating a memorable experience for participants. Hence, without further delay, may I now request our Director, Ma'am, Professor Dr. Anuradha Mishra, Ma'am, to felicitate the first speaker of our first technical session, Professor Ajay Sharma, sir, with a planter. Because here at MIT, we believe that gifting a plant to someone is gifting a whole new life. May I now take this privilege to introduce to this awesome gathering, both online and offline, our esteemed speaker of the first technical session today, either Professor Dr. Ajay Sharma, sir. Professor Sharma is currently working as a professor and head Department of Pharmacology, School of Pharmaceutical Sciences, Delhi Pharmaceutical Sciences and Research University, New Delhi. He has done his PhD from Pune College of Pharmacy, Bharati Vidyapeeth University, Pune, and has more than 18 years of teaching and research experience. Our honorable speaker has been awarded postdoctoral research fellowship from Yunnan University, Republic of Korea, senior research fellowship from Department of Biotechnology, New Delhi, India, and junior research fellowship from UGC, New Delhi, India during his studies. His research areas include drug discovery and development from natural resources, ethnopharmacology, natural product chemistry, metabolomics, plant biotechnology, bioactivity guided fractionation, and bioconversion. He has published more than 90 research articles so far in peer-reviewed journals of national and international review and has presented more than 150 papers in national and international conferences. Honorable speakers has contributed 6 book chapters and 19 book chapters till date and has published over 12 Indian patents so far. To pursue several research activities, he has been bestowed with research grants from reputed funding agencies including IUS, DBT, AICTE, and MPCST. Dear delegates, now I'm also pleased to inform you that our honorable speaker has been listed as top 2% scientist as per the list of Stanford University successively from the last 2 years. We are really blessed to have you, sir. May I now have the honor to invite you on the stage and make your scientific presentation. We are really glad to have you on. Have that. So I would request you to please stick to this podium because you have to be visible online. Dr. Mahapatra, please you offer.
So thank you very much, Dr. Mohapatra. Am I audible to all?
Just stick in video.
Okay, this is.
So you can
It is not coming. Okay, 45 minutes. Yeah, definitely.
Thank you for the phone call.
चेंज तो यहाँ से भी कर सकता हूँ.
No, sir. It is fixed for the podium itself. So they will do next. You can just call for the next slide.
ठीक है.
Thank you. Thank you, sir. Please start.
So good morning to one and all present over here. First of all, I would like to thank the management of this beautiful campus, MIT University, Lucknow, and the director, Professor Mishra is there, the organizer of this conference, Dr. Abhishek and Dr. Versa. They have invited me and given me a chance to interact with you people. Before going to this particular topic, I would like to tell you my association with the MIT University Gwalior. I have started, I joined MIT University Gwalior in the year 2019. And I was the founder member over there when that particular campus was started, MIT Gwalior Pharmacy. So I joined with Dr. Nagappa. He was the director over here. And I am connected to this campus as well, though this is my first visit over here. But your current VC, Pro VC, Professor Vasist, we worked together over there and we were sharing the floor. So I was working in the pharmacy and he was heading the department, that management, AIB over there. And before Professor Vasist, I think Professor Dhaneshwar was also there in this campus. So Dr. Sunila Dhaneshwar and Dr. Sunil Dhaneshwar, both of were my teacher when I was pursuing my PhD degree. So I am having, good association with MIT University. So just I would like to start my topic. If you see the title of this topic, Unlocking the Power of Nature, Recent Breakthrough in Natural Products. First slide title. Recent breakthrough in natural product research. Before starting to this particular topic, you can see or you can compare our Indian system of medicine with the Chinese system of medicine, and why today's all are or all want to working in the area of natural products. Because the natural product, they have hidden potential, and that particular potential we needs to explore. Clear? And if you compare our Indian system with the Chinese system, the Chinese system of medicine is well documented and scientifically well validated as well. But where we are lacking is the lack of scientific documentation and validation. And that's why the Indian government, or particularly if you see the Ayush Ministry, they are giving huge funding in this particular area of research to unlock or harness the potential of natural products. I was discussing with my colleagues. So one is from pharmaceutics, one is from pharmacology. Just when I was, taking that breakfast over here, they are all working in the area of natural products. So natural product, it solely doesn't mean pharmacognosy, because nowadays, if you will see, the trend is moving towards interdisciplinary research. So whatever the branch, whatever the, departments you are having, somewhere directly or indirectly, the researchers or the scientific community, they are dealing with the natural products. So that's why this particular area is very, very important to see and to explore. Next. Now, all of you are aware about the basics of the herbs and the herbal drugs, but natural products are more than that as well. So here we are not only dealing with the plant drugs, we are dealing with the, plant extracts. And recently, some new concept also came in this particular area. That is the concept of phytopharmaceuticals. Okay. And the peoples, they are also working in the area of herbal formulations. So not only we are dealing with the plant related drugs, we are also dealing with the metals, mineral drugs, as well as the combination of herbs and minerals. So that is basically the beauty and the, importance of herbal drugs as well as the natural products. And these are the drugs which are obtained from the basically plant resources. and they contains n number of phytoconstituents. So the beauty of this particular area of research is, n number of phytoconstituents are there if you see any of the herbal drugs or any of the prepared extracts. Next. Now, before going to this particular topic, I would like to touch upon what revolutions happened in the area of natural product research, or you can say the herbal drug technology. How we have started. Next. Now, all were watching Ramayana in our earlier days. So the concept of drugs came that time only, even before then that also. And when the Lakshmanji ka case, all of you were knowing, when he was hit by Meghnad, and how you can see the Lakshmanji, they brought the Sanjeevani booty, there were Ved, that was Susain Ved, and how he saved the life of Lakshman Bhagwan by simply giving the extract of that Sanjeevani booty. So the concept as well as the applications of herbal drugs was there, but there were no regulations, there were no standardization. The doctors or the Vaidyas who were available over there, they were simply preparing the extract and they were giving or administer that particular extract to the patient itself. But if you see, there were no regulations, there were no concepts of drug and cosmetic acts. The peoples were using and the knowledge that were written in various ancient books and various, Vedas. The Rig Vedas is there, the Athar Vedas is there, and n number of ancient holy books were there where the description of herbal drugs were included. And if you will see the second revolution which has been happened in the area of herbal drug technology, that is related to the herbarium preparations and the ethnopharmacology. Peoples were having traditional claims. There is only one need, you have to validate it on scientific basis. So these were the two revolutions. Next. You can see the third revolution of herbal drug technology, the concept of preparing herbal extracts, the concept of electronic microscopy to visualize or to see the cellular structure came in existence, the preparation of, various plant extracts, the isolation and the characterization of phytoconstituents were there, and the concept of high throughput screening came. Because whenever you are dealing with the herbal drugs, sometimes or most of the times we are blank. We don't know how we have to move further, which particular extract we have to prepare, because n number of extraction protocols are there, n number of phytoconstituents are there. So sometimes we are blank which particular technology, which particular, solvent we have to deal with. So in that particular case, generally we are preparing numerous extracts starting from non-polar to the polar. So you will be having, a range of plant extracts, and, gradually you have to explore their pharmacological potential. Next. Now the concept of fourth revolution came again. So here you can see the concept of plant tissue culture. That is one of the very, very important area of research where you people can build your career or you can go for entrepreneurship. If you will see the social sites nowadays, n number of persons, they are putting some videos, some clips, how they are cultivating the mushrooms, how they are cultivating kesar in the area of 100 by 100 or 100 square feet or 200 square feet area. So this is basically one of the very important area of research, which is directly deals with the conservation of the medicinal plant, because conservation of medicinal plant is the need of our, as we are basically dealing with the species which are endangered in nature. And if you like to increase the concentration or the percentage of phytoconstituent in the plant material, you can take help of this important concept. Now, another concept which came during the fourth revolution, that is plant biotransformation, because in the chemistry, we have read n number of, the reactions, how we can move from A to B or B to C or C to D. So like this way, n number of biochemical reactions are there even in the plant kingdom, by which plants are synthesizing primary and secondary metabolites. So you can use the plant as well as the other, biological potential of the plant you can use to convert, a particular substrate to the particular product. And then you can see the applications of laser scanning, confocal microscopy, electron micrographs, as well as the concept of reverse pharmacology came, particularly you see during the COVID times. Next. Now, this is the post-COVID revolutions. You see, during the COVID times, we were not having any the drugs which can combat COVID-19 virus. So we were getting some information from different resources or sources, and accordingly we were, trying to combat that particular virus. So during the COVID times, the concept of, IU genomics came. And if you see the COVID time, after every 15 days or 1 month, there is another COVID virus. So kabhi Omicron aa jata tha, kabhi koi dusra virus aa jata tha, because, very frequently the mutation were there. And in that particular case, it is also very difficult to develop a particular remedy against that particular virus. So in that particular case, what the government has taken the initiative, they just utilize the concept of IU genomics, how we can deal, the mutation or the, gene with the concept of Ayurveda. And during the COVID time, our university, they came up with one of the important formulation, that is chloroquine zinc. So, I would like to, discuss one of the challenge with respect to that herbal formulation that we have developed. We did not get, Ayush approval, because we want to, market this particular formulation as a drug. But that particular drug, we have developed in hurry, just like a vaccine, you can see. And we tried to contact Ayush Ministry, but they told we would not give you the drug license. So finally, our authority, they have decided why not to launch this particular formulation as a nutraceutical or as a food, where the regulations are quite relaxed. So we have, marketed this particular formulation as a nutraceutical, and you will be fortunate or you will be surprised. Okay, this drug is in market in US as well as the UK kingdom. And you can see the importance of the herbal drug when the TKDL establishment is there by the Indian government, because WHO, they have recognized the potential of herbal drug. So WHO even, they came forward and they understand our ancient Indian system of medicine, and they are now funding in various area of herbal drug research. So these are some of the revolutions that happened or that took place in herbal drug research. Next. Now you can see the 21st century where we are, and here these are the 3 important technologies which may give us the solution. That is the biotechnology. You can see the vaccine development during the COVID times, when 15-20 years minimum is required for drug discovery and development, and how fast we were that time, how the vaccine were developed against that COVID-19 virus. Though some of the studies we have missed during that time because of, urgency or emergency, and you can see what type of, the side effects we people are facing after, vaccination. So, the ICMR, they are conducting some of the research why the cases of, sudden cardiac failure or cardiac arrest is coming up. So that is basically the biotechnology. Herbal drug technology I will be touching upon. And the concept of bioinformatics is again very, very popular, especially in the abroad. If you will see, if you are aware about the concept of, AI, the IT, and if you want to, read the information, how the information will move from DNA to RNA or proteins or post-translational modification, you will be having, bright career. Next. And if you will see the herbal drug technology, there are various terms which are popular, like nutraceuticals are there, cosmeceuticals are there, skinceuticals are there, biopesticides. So in one or another way, the peoples or the scientific community, they are working in this particular area. This is the figure. Around 88% of the world population, they are based on plant-based natural products. Next. Our Indian system of medicine, it is very old, and we have Ayurveda, Unani, and Siddha. And all these complementary and alternative system of medicine, they are easily accessible, affordable, and effective. And if you will see the herbal drug market, by the year 2050, it would be around 5 trillion US dollar. So this is the figure, which can attract young investigators or the researcher or the entrepreneur to come forward in this particular area, and you can develop your area or you can develop your future. Next. Now you can see we are rich with biodiversity. Our country, if you will see, we have, 2.4% of the area of the world, and 8.1% of the world we are having biodiversity. And if you will see the Indian plant species, they are around 45,000, along with, 35,000 Indian Ayurvedic formulations are there, along with bhasmas. So you can see the number. What is the problem with the Ayurveda is most of the formulations, they are not explored with respect to the active phytoconstituent what is inside. So if you will see any of the Ayurvedic formulation on the label, if you will see, they will be writing ashwagandha this much percentage, ginger this much percentage, turmeric this much percentage. But what about the phytoconstituent, which are the main bioactive inside that formulations? So there is a need, to work in that particular area. Next. And if you will see the industries, we have very small scale to small, medium, and large scale industries depending upon their turnover. And among all the systems of Indian medicine, Ayurveda contributing maximum, followed by homeopathy, Unani, and Siddha. You will be surprised. We hardly know the concepts or as well as, the applications of Ayurveda, little bit followed by nowadays, the peoples, they are moving towards homeopathy, but very less peoples, they are aware about the importance and as well as the applications of Unani as well as Siddha. So if you will see the Ayush ministry, there are n number of various departments they are dealing with, like for Ayurveda, we have the CCRAS, but for the Unani and for the Siddha and homeopathy, there are separate department who is encouraging young investigator to submit the proposal and take the grant from them. Because these are very, very less or least explored area of research. If you will see the global herbal market, it is around 62 billion US dollar. Next. Growth potential, 15% growth potential, annual growth potential is 15% approx is associated with this particular area. And you see there are n number of listed herbal companies which are the major player in this particular area of research. Most important example is Patanjali. You can see what type of products they are bringing to the market now. If you will see the noodles, that is also coming from the Patanjali. If you will see the Maggi or the ghee or the butter or the milk products, they are coming up with n number of things where they have the scope, they have the business potential. Next. Why the peoples are looking for the herbal drugs? Because they may give us the solution in case of primary healthcare, the infectious disease, the degenerative disorders where the modern medicines, they won't give you that much, support. The metabolic disorders, particularly diabetes, we are the hub or we are the capital of diabetes. You can see in each and every family, diabetic patients are there. And if you will see the modern system of medicine in diabetes, what is there? We have very limited medications in the modern system of medicine, but they can't cure the diabetes. They simply just manage the glucose level and other symptomatic treatment is associated. But if you will see the herbal drug research, the chances are there, the scope is there. Next. So why herbal drug? Why we are talking that much? So there is one common assumption or perception. They are comparatively safe because of huge margin of safety and less technical input is required to administer these drugs. The peoples are seeing increasing complexity with the modern medicine and they are expensive and they are associated with the side effects as well. Next. Now the person who wants to work in this particular area of research, they should be having, multi-dimensional skills. They should be aware about the basic concepts of botany. That is very, very important because where we are lacking in herbal drug research, that is the standardization. We won't be, we can't identify the particular herbal drug easily. And from where we are procuring the herbal drug, that is the Asian biggest market in New Delhi, that is the Khari Babri. If you will go there and if you will see the condition of herbal drug, you will become mad because they are drying the herbal drug on the floor itself. And if you are mixing herbal drug 2 or 3 together, and if you are making it powder, it would be very difficult to identify where is ashwagandha, where is turmeric, or where is other, because they may lose their identity once they come in powder form. So the scientists, whatever, if they are the pharmaceutics expert or if they are the biotech expert or the pharmacology expert, they should be having that much knowledge of each and every, domain, the botany, the ethnobotany, the chemistry, as well as the pharmacology. Next. This is very important slide, you can design your research work and you can, build your whole career using the concept of this slide. How the drug discovery and development has been taken place, from where we have to start. So if you want to, build your career in this particular area, the first and foremost important task is collection. Just 2 or 3 days before, we were having, interaction with one of the important pharmaceutical company, that is Amil. So we were talking how the students get placement in your company. They were telling we don't need any skill. If they are able to identify the herbal drug only, we will give them job. Because, the identification of the herbal drug is a really big challenge. And if you will see our PCI curriculum, earlier in our time, we were having, the concepts of botany when I was studying B.Pharm. But if you will see nowadays, there is no botany type of concept in pharmacognosy. Everything is totally missing, which is the first and prime step in herbal drug research. So whether you are able to identify or to pick the correct herbal drug or not, because everything will be depending upon them. And nowadays, the concept of adulteration and substitution is also existing. So that may, increase the headache of the researcher. So first and foremost important part is the collection of the herbal drug. And to collect a particular herbal drug, you have to refer literature. That may be our ancient books, that may be the scientific publications which are available. So sufficient, literature survey is required. And after that, you have to take help of botanists and taxonomists to identify it. And once they will give you the green signal, then you can move for the drying part. And there are n number of drying techniques. You can dry herbal drug by any means because hundreds of means are available. But again, you have to be, very much careful while selecting the methods of drying. That may be shade dried, that may be sun dried, that may be oven dried. So it depends upon, the nature of your drug as well as the nature of the phytoconstituent what you are going to target. And then the extraction. So once we are talking about the extraction, always the idea came, okay, soxrate laga do ho gaya, maceration kar lo, percolation kar lo, because only these techniques we know. And that is generally, mentioned in our routine curriculum. But are we knowing there are some green techniques of the extraction that is environmental friendly and that increase the yield of phytoconstituent in the particular plant extracts? So nowadays, the peoples or the scientific community, they are using those techniques as well, and they are microwave assisted extraction or, the sonication. And after extraction, you will get the crude extract. Again, it is a, big headache because if you want to purify it, you have to be well versed about separation and purification technique from normal methods of purification and separation to the sophisticated technique like preparative HPTLC is there, LCMS, LCMS-MS, and other HPLC is also there. So like this way, you have to be well versed aware about the separation and purification technique. You can start with TLC in the laboratory because that is the basic techniques, and later on you can use the sophisticated analytical techniques as well. So by this way, you know, you will be aware about the fingerprinting profiling of that particular herbal extract. And suppose if you are able to isolate the single phytoconstituent, you have to identify it. And for identification, you can start your journey with the elemental method of analysis up to the NMR and mass. So like this, you know, the techniques are involved for the identification of single phytoconstituent. And suppose if you are identifying particular phytoconstituent, then you should be ready for the biological screening. So you can go for the clinical screening from phase 1 clinical trials to phase 5 clinical trials, and then go for FDA approval, and then you can market your drug. So like this way, you know, various steps are there which are involved in drug discovery and development. And nowadays we are having this concept as well in our laboratory, that is bioactivity guided fractionation. So if you are preparing 10 or 100 of the extract, you can, you know, check those particular extract for their biological potential. So which one is, you know, giving you more activity, that you can select and you can proceed with that particular extract further. So that is the bioactivity guided fractionation. Next. This is again one of the area, the in silico based. Dr. Abhishek is there, one of the expert and the collaborator with me. We have submitted, you know, one research project to IOS, and that is funded as well, around 80 or 85 lakhs, Abhishek, like that. So, you know, we have included the concept of, you know, molecular modeling as well, because, you know, plant contain n number of phytoconstituent. You can, you know, target those particular phytoconstituent against a particular target where they are showing maximum binding affinity, that you can select for your study. So that is basically one of the method we are basically we are dealing with. Next. So why the standardization is important and necessary? Because we don't have, you know, scientific validation and documentation of the herbal drugs. So that's why standardization of the herbal drug is of prime importance. And that's why our maximum products or the herbal drugs, they are refused by the global agencies because of lack of scientific documentation. What are the various advantages? If we have the set protocol for the herbal drugs as well as their extract, we can improve industry institute linkage. We can assure the quality assurance. We can assure batch to batch consistency of our product, as well as the acceptability of our product can be increased manifold. Next. There are various agencies, various guidelines are available to standardize the particular herbal drug as well as extract. So FIP, WHO, German Commission, you can pick. Generally in our laboratories, or we are following WHO guidelines for the standardization purpose. Next. So what it means, it will ensure the quality, safety, and the efficacy of the herbal drug as well as their finished product, which is the need of our. Next. So how to standardize? First, very important task in standardization. If your raw material standardization, half of the journey will over. Okay. And then followed by the process and the product. So you have to standardize the raw material, followed by process, and then the product. So for raw material, if I ask you, there are n number of factors that affect the quality of the raw material. And of course, in turn, it will affect the quality of the finished product as well. So the season, okay, if you will eat mango now, you won't get that taste which will be available in May or June or at the peak time. Okay, so season affect, you know, the accumulation of phytoconstituents. So followed by the reason, of course, Kashmiri mirchi very famous hoti hai. South Indian spices, they are very famous. Okay, so the reason, of course, it affect the quality. of the finished product, then the time of collection of that particular, herbal drug is very, very important. The drying method, the preparation, and the storage. So how we are storing herbal drug in our kitchen also, if you will see, generally we are using some containers and we are putting herbal drug inside it. And sometimes the drug is not properly dried as well. What will happen after that? Fungal infection, fungal growth, fungal contamination. Okay. So like this, you have to be well versed about the storage techniques. These are the various, important parameters that you have to evaluate for each and every drug before moving forward. And they are the heavy metal count, microbial count, chromatographic, profiling, the presence of foreign matter, because sometimes if you are, drying herbal drug in an open area, you may find insect excreta as well inside it. Okay. So you have to be, very careful. Sensory evaluation. If you will go to the market, if I ask you, okay, bring some cardamom, कैसी खुशबू वाली इलायची मिलती है? बिल्कुल exhaustive इलायची, exhaustive clove you may get it from the market, because what is the phytoconstituent or important bioactive in those particular drugs? They are the essential oil, and most of the essential oil already it was extracted, and the exhaustive drug is available in the market. The ash value, the bitterness principle, the foaming and swelling index is important, and the radioactive contamination, particularly in the area if they are, if the radioactive, radiation is there, you have to identify, and the pesticidal residue. Next. So I can give you some of the research output. This is the drug which were, available in the Western Ghats of the India. The molecule, camptothecin, topoisomerase 1 inhibitor, and it is very effective against solid cancers, particularly breast, uterine, and cervical. So we have started our journey with the microscopic study of that particular herbal drug. And like this, you can see these all are hand-drawn images using camera lucida. Next. You can see the TS of the petiole of Nothapoditis plant material. So you can see how the cellular arrangement is there, how the cellular structure looks like. Next. This is the midrib ka TS of Nothapoditis. Next. This is the lamina TS. So you can see how the TS has been performed. So all these data are very, very important, to identify adulteration or substitution in a particular herbal drug, and they are the part of monograph development. This is the quantitative microscopy of the leaves. You can see various, the cells are there, and we have quantitatively analyzed. Next. The physicochemical parameters. So we all are aware about the ash value, but it is not the first and last. There are other parameters as well that you have to identify: the loss on drying, the foaming, swelling index, as well as the electrical conductivity. Next. The extractive value. So various extractive value we can perform, and that is specific for a particular drug. Next. The characterization, whatever extracts we are preparing, each and every extract you have to characterize for their color as well as consistency, and that is again very specific. Next. The metal analysis that you can again analyze. This is again very, very important task because a number of heavy metals are accumulated. Next. The fluorescence behavior, again it will give you idea what type of extract or the preparation is. You can see we have to observe the fluorescent behavior under visible light, short UV, as well as the long UV light. Next. There are various observations of the plant powder with a different reagent if you are treating it. So that each and every observations you have to note down. And this is the TLC or the HPTLC profiling of the leaf extract. So by this way, the number of spots indicating maybe that many number of phytoconstituents are there, but this is the rough data. Again, you have to work on that. Next. The process standardization. So process uniformity must be there, where you have to optimize, the material, the schedule of the mixing, the time, temperature, and the humidity, and in process quality control parameter, the determination of pH, moisture, and viscosity content must be there. And whatever the formulation or the preparations we are doing, there must be batch to batch consistency, reproducibility, and repeatability. These are the quality assessment parameter for the herbal formulation. So there are n number of parameters that you may use it. It depends upon the type of the formulation, whether they are tablets. So you will be having separate parameters like weight variation, shape, and size. And if you are having oily preparations, there may be separate parameters for acid value, saponification, or ester value. This is the fingerprinting profiling. Like this, you can, process it to identify the presence of marker compound. Next. You can see how we have identified the presence of jad, guggulsterol, and guggulsterone in guggul extract, that is commiphora. So you can see below is the TLC fingerprinting profiling, and you can see the densitogram as well of the bioactives of guggul. Next. You can see the TLC profiling of Sallaki, which is important anti-arthritic drug. Next. This is the TLC profiling of Lesia. So that I took from Dr. Arya. Their PhD work is there. So how they have screened different extract to check the presence of phytoconstituents. Next. You can see there are different chromatographic technique, how we can use them to identify the gallic acid from Phyllanthus embolica. You can see the HPLC chromatogram. Next. This is the, HPTLC profiling of Nothapoditis. So the third spot on the top line, you can see that is, fluorescent blue. That is an indication of standard compound. No, that standard compound is at J. And all, if you will see, they were the different samples and their RF value is resembling with each other. It is an indication of that all the samples, whether they came from natural source or the in vitro source, they may contain that phytoconstituent, which is anti-cancer camptothecin. Next. So this is the HPTLC profiling of Lesia spinosa extracts. Next. How we can, performing experiment in our laboratory. So first we have to, draw the calibration curve. And once you will get it, then you can go for the quantification of the particular bioactives. You can see the HPTLC densitogram overlay of camptothecin in natural and tissue culture grown plant material, how the O spectra overlay is there. And this is the HPLC chromatogram. Next. So the geographical variations we have found over there, the particular plant material that was collected from 4 different locations and 1 location accumulate maximum amount of anti-cancer metabolite. So this is an clear indication of geographical variation of the bioactives. By this way, we can do the UV spectroscopic fingerprinting of the Chamanpras, which is available in the market. So see how the spectral overlay is there. It means they are having in particular phytoconstituent in all the sample tested in the laboratory. Next. You can see again HPTLC spectral overlay. Next. The GCMS. So if you are dealing with the terpenes or the essential oil type of, component, there is one technique which is available. So you can go for the complete GCMS profiling of the essential oil. One beauty with this technique is the library is available with the system. So you can use those particular library and you can identify the particular phytoconstituent. Next. This is another parameter. Before going, further, you have to identify or assess the safety and efficacy. So where you will be using the concept of general pharmacology, the toxicology, as well as the clinical trial. And you have to check acute, chronic, as well as the long-term toxicity. If you are developing the herbal formulation, then definitely stability assessment must be there where you have to use US FDA guidelines. So that is for accelerated stability study. Next. These are the various monographs that you may refer. Next. Some of the limitations are there. If you are collecting the plant material from the natural habitat, always there must be the limitations, because n number of factors are there that affect the accumulation of phytoconstituent. Maybe the reason related, maybe the time related, maybe the other geographical factors. Okay. So some limitations are there with the conventional process. Next. So to minimize the effect of those external factor, we are having some another approaches by which we can standardize the herbal drug, and that is the biotechnological approaches, where you can use the concept of DNA based molecular marker. This is again new area where the government funding agencies, they are offering funds. Okay. So if you talk about the herbal drug, okay, we are evaluating their pharmacological potential. We are, developing their analytical profiling. There are a number of scientists, but very few of them, they are basically working in this particular area, how the complete genomic profiling can be developed for a particular herbal drug. Because genome is very stable in nature, and this will be very helpful to identify the chances of adulteration, which is one of the biggest challenges. And another one is micropropagation. So what type of plant material you would like to develop in the laboratory using in vitro technique, depending upon your objectives, you may develop, because you have the technique in your hand. Next. So various, DNA-based molecular markers are there. I do not want to go in details. This is the application. You can analyze the whole genome, and you can study the phylogeny and the evolution aspect of plant material. And you can go for the mapping and labeling of particular genes which are involved in the, biosynthesis of primary and secondary metabolites. These are the various molecular markers. You may opt either of one depending upon your objectives. Various steps are there. Next. This is the proposed DNA isolation protocol you may go for. Next. Now the application, using the concept of molecular marker, we can identify adulteration or substitution among very closely related species. Like Panas ginseng, it is oftenly substituted by American ginseng. So how you will identify, because the species are morphologically, they are closely related. Phytochemically also, they are very closely related. But genomic wise, they are different. So you can take help of this particular technique, and you can also standardize the important formulations which are available in the market, like Tirfala Churan, बहुत ज़्यादा हम use करते हैं आयुर्वेदा में. So you can use this technique to identify it. These are the some of the genome sequences of the medicinal plant. Next. So now the genome driven drug discovery is also there, where we are using the concept of molecular, just like we are using the concept of molecular modeling in genomic driven drug discovery, we are using the concept of network pharmacology. So you can identify the particular phytoconstituent which are targeting on particular genes or the proteins, and accordingly the drug discovery may be proceed further. Next. So I hope so. Maybe this may be the last slide. The micropropagation. So as I told you, the plant material, they are affected by, n number of external factors, which are environmental most of the time. But if you are developing that particular plant material inside the laboratory using biotechnological approach, you may, overcome the effect of external factors. So for that particular one, the plant tissue culture is one of the approach. Next. So this is the basic concepts. You know the concept, what we have studied during 11th and 12th class, the use of auxin and cytokinin, what they favor. They are the rooting and the shooting hormones. Okay. Next. And this is the micropropagation of Gymnema. You know the application of Gymnema. This is very important anti-diabetic plant. And, fortunately, we have grown this particular plant in the laboratory. And you will be surprised, we could increase the consent of active principle inside the gymnoma, which is considered as very important anti-diabetic drug. Next. How we can proceed. These all are the, just like test tube baby, you can see how the baby can be grown inside the laboratory. So these are the plant babies. You can start with the callus initiation and you can end up your experiment with the suspension culture, the metabolomics and the genomics. Next. So hairy root culture, I think all of you are aware about this technique. So where the genetic involvement is there, these are the root of ashwagandha, because you know ashwagandha contains active principle inside the root, that is vidonolide or others. Okay. So how we can grow it inside the laboratory using agrobacterium species. Next. These all are natural images, not from internet, from our PhD studies. So how we can, go for the suspension culture of those plant material, you can propagate or multiply it using liquid culture. Next. Then again, rapid multiplication, you can change the percentage of the hormone. Next. Hardening. Now the plants are ready because now they are accustomed to the environmental conditions. So initially you were growing them in the greenhouse, and later on you can transfer those plant material which are test tube baby of yours, you can transfer to the field. Next. So thank you very much. This is my whole research journey. If you are having any questions, please, you can ask.
Thank you, sir, for this elaborative session of yours. Now the forum is open for an interactive session. If we have got any questions, I think we have a couple of questions in the chat box from online participants. Sir, if you can address the same, I'll read the question for you. Also regarding extraction of polyphenolic compounds, usually they are heat sensitive. So any particular suggestion regarding the extraction of these class of compounds? Question is from Nabaru Mukhopadhyay.
Yeah, there are n number of techniques. So the hot method of the extraction is there. The cold methods are there. So in hot, the Soxlet is there. Generally, we are using in the laboratory. But if you are having, the heat sensitive compound, or in case of herbal drugs, sometimes we don't know what type of components are there, whether they are heat sensitive or not. So in that particular case, generally what we are referring, just go for the maceration. Or if you want to opt the environmental friendly technique, you can go for the ultrasonication as well as the microwave assisted extraction technique. So it is not only related to the polyphenol, because you are targeting the extract, and in extract only polyphenols are not there. Some other, important heat labile constituent are there that may show synergistic effect along with the polyphenols. So rather than doing A for but, just I would suggest you go for the maceration or go for the environmental friendly technique, that is sonication or the microwave assisted extraction. And in these technique, there are n number of reports which is indicating you may get better yield of the phytoconstituent rather than typically Soxlet or other methods. Yes.
I do hope that Navarun has received his answer. We can have another one question online. I think it is again from Navarun. Again another question from Navarun regarding herbal raw material standardization. Geographical variation plays an important role, I suppose. So can you give some suggestion regarding, how geographical variations affect the constituents?
There are n number of, again, research paper which is clearly indicating there is the direct correlation between the geographical factors on the accumulation of phytoconstituent. As I told you in my slides as well, I was targeting camptothecin, which is anti-cancer compound. And initially, I thought I have to collect that particular plant material from Mahabaleshwar. You know that place in Pune. near to Pune. Mahabaleshwar is there. So there was one site we have identified where the plant material was available in plenty. We went over there, we have collected the plant material, and what we thought is if geographical variation is there, then monthly variation may also be there. So in a year, because I have completed, PhD studies in 3 years. So 36 times I visited Mahabaleshwar, not only one times. So like this 36 time on monthly basis, that monthly 12 month of, for example, 2007, 12 month of 2008, and 12 months of 2009. So like this 36 month were there. I have collected the plant material from the same place, and I have, analyzed the concentration of camptothecin in the plant material. So you will be surprised. There is entire, variation is there. There were particular month where the accumulation was maximum, and that was in the month of December. So that I am giving you the example how the monthly variation is there. Now you come to the geographical variation. Initially, I was targeting Mahabaleshwar, but I came to know there was another place where the plant was also available. So in Maharashtra, there were other places which were 150, 200 kilometers away from that particular spot. That is Sirsi, Patan, Kolhapur, aapne naam suna hoga. So I visited those places as well. Not that many number of times, but I have collected the sample and I have compared the result of all the samples collected on geographical basis. So there were, clear-cut indication of geographical variation, monthly variation, as well as the variation among the different plant parts. Because the plant was bearing the flowers, the fruits, the roots, as well as the stem. In fruits were also, some fruits were immature and some were fully mature. So even, within the same plant material, there were 6 or 7 samples I have prepared, and the result clearly indicating there is sure sort variation among the particular phytoconstituent. Very simple example, you are using kitchen spices. Very simple. Some spices which are available in the market, they are aromaless. You won't get any taste out of it. But similarly, if you will use South Indian spices or Kashmiri chili, I think you will become fan of that. So that is basically clear indication of accumulation of bioactive principles.
Thank you, sir. Due to constraint of time, we will be sending you the next couple of questions. Maybe I would request you to please be here with us online and on the stage as well. Thank you for your insightful and engaging presentation, sir. And I now request Professor Dr. Anuradha Mishra ma'am, Professor Dr. Abhishek Tiwari sir, Professor Dr. Harsha ma'am, Dr. Nisha ma'am, to please join us, and also Professor Dr. Devu Prasad Kanda sir, to please be here on the stage to facilitate the speaker. Can I request you to please come out on the stage? I also request the student volunteer with the memento. For the members to join their hands to felicitate the honorable speaker with a memento as a token of appreciation and thanks from LT Institute of Pharmacy, LT University of Lucknow campus. Thank you, sir. We are grateful again that you share your expertise and enthusiastically engaged our audience. Thank you for taking your time, support, and expertise and be here with us for this wonderful session. Plant.
Respected dignitaries and my dear delegates, I am proud enough to announce here that we are now having among us the esteemed speaker of the second technical session of Day 2 RDPS 2025, Professor Dr. Vibhu Prasad Panda, sir. I would request our Honorable HOI Ma'am, Professor Dr. Rana Mishra, to kindly felicitate the esteemed speaker with the planter. As sir is also from pharmacy and phytochemistry, and here I am admitted we believe that presenting a planter to someone is giving them with a new life. And I do hope that this planter will remind sir about today's session of this. Thank you, ma'am. I'll just take a few minutes of yours, sir. If you can please be with us on stage to just officially make an introduction of yours. I take this privilege now to introduce the Honorable distinguished speaker of this session, Professor Dr. Vibhu Prasad Panda. Professor Dr. Vibhu Prasad Panda is a distinguished researcher with more than 22 years of experience in nutraceutical and bioprocess development. He has secured 13 research grants from prestigious funding agencies, including DST, DBT, UGC, DHR, and ICMR, totaling worth rupees 4.2 crores. As a consultant to core Indian nutraceutical and natural product industries, he has also successfully transferred fermentation technology for astaxanthin production to Beijing Global Biological Corporation, China for $50,000 international technology transfer deal. Few of his imperative technological innovations also include rape yeast rice, yeast astaxanthin, soy isoflavone, vitamin K2, MK7, phycocyanin, and phytopharmaceuticals for bone health. Professor Panda has authored more than 109 international research publications till date and holds 2 patents on bioreactors and plant metabolites. He has guided over 15 PhD scholars and 53 PG students till date, revealing his true research potential. Thank you, sir. Your presentation is definitely going to be thought-provoking. And now the stage is all yours.
Thank you. Good morning, all of you. Sorry, good afternoon, all of you. Because I thought it was before 11. Okay, now today we are going to discuss about the natural products, right, and how we can develop new natural molecules or natural products by using the bioprocess technology or by using the biotransformation process, right? That means how we can use the biotechnological tools to develop new drug molecules from natural origins. Clear? Now, topic of my talk is biotransformation of natural products. All of you know what is natural products, right? Whatever the molecules we obtain from the plant or microbes or any of the natural source, right? We call it natural product. But if a molecule we get right directly from the nature, right, how we can change it to new molecules? Those things we will discuss today. That means development of newer phytomolecules through biocatalysis and biotransformations. Now, what is biocatalysis? All of you know catalysis process, right? When you use enzyme to convert one molecule to other molecules, right? We called it biocatalysis, right? And when we use the live cells to convert the molecule from one to other, right, that we call the biotransformations, right? Sometimes biotransformation are also known as metabolism, right? In pharmacology, you might be knowing that biotransformation means metabolism. But if you in the point of natural products or in point of biotechnology, we actually biotransform means conversion of one molecule to other molecule by using the live cells. It's not the metabolism. Now, when we are discussing about the biotransformations, right, mainly we will use the microbe, right? Microbe is the source, right, where we can use the microbe, right, and you can convert the molecules, right? Therefore, you know, the microbe is the powerful living cells, right, in the world. That's why Louis Pasteur already told that never underestimate the power of the microbes, right? These are okay, never underestimate the power of the microbes. That means they can do anything, right? They can be trained to develop a new microbe, right? And then they can do anything what you want. Okay, they can cause disease, right? And they can also produce some valuable molecules. How we are using the microbe, that is depend upon you. Now, what is actually the concept of biotransformation and developing new one? Now, whatever the drug or natural product or molecules we take, right, actually it is transformed in our system. Under in vivo conditions, actually they are transformed, right? Particularly in the gut. In the gut, actually there are some microbes. We called it microbiota, right? These microbe actually, they convert the molecule to one to others. But we don't know what type of molecule they produce inside our gut. We also don't know what type of microbe present in our gut which convert the molecule from one to others. But if we understand the microbe as well as the molecule which is formed and which enzyme is responsible for converting the molecule to others, right, then we can develop new drug molecules, new natural products, new phytomolecules. Got my point? Is it clear? But sometimes we actually don't know what product actually formed in our system and how they are actually useful pharmacologically, right, and bioefficacy, right, and how they are helpful in the increasing the bioavailability of the drug molecules or natural products. Again, right, that means we can have chemocatalysis mode, right, to convert the molecules, right? We can also have a biotransformation mode, that means using the live cells, and also we have biocatalysis by using the enzymes. But we have to do research, right, on these aspects. But biocatalysis and biotransformation is the part of biotechnological work, or particularly pharmaceutical biotechnology peoples can work on this area. Chemocatalysis can be done by the chemist peoples. Therefore, we will more focus on the biotransformations and biocatalysis. What happens here is that if you are knowingly converting a molecule to others, that means that is called the targeted biotransformation. That means the target is known, right, and you are converting the molecule from one to others, right? Then that is called targeted biotransformation. And non-targeted biotransformation means whenever you are taking a molecules, right, and transformed by the cells, okay, to different molecules, then that is called the non-targeted biotransformation. That means you don't know the target, but the molecules are a mixture of molecules. Clear? Now, what is the concept here? Substrate. Substrate means the molecule, right? Phytomolecules, original phytomolecules, which you obtain from the herbs. What Ajay sir already discussed in our previous talk, right? The substrate means it is a natural product, molecule, right? But when you culture the substrate in presence of a cell, cell means living cell, right? Probiotics or any microbes, right? Then what happens here is that the cell convert the product into different metabolites. Metabolite, here I have shown you three metabolites, metabolite 1, 2, or 3, right? It may be n number of metabolites produced by the same cell because the cell contains a lot of enzymes, right? When you feed a drug molecule to the cell, it converts into other molecules, right? These are called metabolites. Now, sometimes what happens, these metabolites are actually, they are highly effective in compared to the original substrate. That we have to understand. What type of metabolite are produced, right, when we feed the substrate to the cells? That means we can develop the new molecules, right, in the form of metabolites. Is it clear now, the concept? Now, next, now if you are doing the biocatalysis, the previous one, that is a biotransformation. Now, in case of biocatalysis, only you will use the enzyme, a target enzyme, right? And that will create only one metabolite. It will not produce number of metabolites. That means if you are producing a targeted metabolites or targeted product, then we have to use the enzymes, specific enzymes, right? And if you have not targeted any metabolites, right, that means you can use the biotransformation process. One example, right? All of you know Glycyrrhiza glabra root, right? It contains a molecule called glycyrrhizin. All of you see this glycyrrhizin molecule. This is a glycosidic molecule, right? All of you know what is glycoside molecule, right? Now, but the glycyrrhizin, when it is transformed or when it is metabolized, it produces 18-beta glycerinic acid. You can see the structure of the 18-beta glycerinic acid. Also, will get the nucleonic acid. What happens here is that when a microbe is allowed to grow in presence of glycyrrhizin or muleae, or we call it Glycyrrhiza glabra root, right? It converts the glycyrrhizin into 18-beta glycerinic acid. Now you can see the structure difference here. Now previously it was a glycosidic molecule, but out of this glycosidic molecule, we have created a glycol molecule by using the biotransformation process. Clear? But when you see the bioefficacy of the glycyrrhizin and 18-beta glycerinic acid, why difference you will see? Glycyrrhizin is actually, it is a large molecule. It is a aqueous solubility. That means a more hydrophilic molecule, right? But when it is converted into 18-beta glycerinic acid, right, it became more lipophilic. Okay, drug likeness property is increased in case of 18-beta glycerinic acid and wide application of 18-beta glycerinic acid. If you see the bioefficacy of the glycyrrhizin and 18-beta glycerinic acid, it is far better than glycyrrhizin because the lipophilicity, lipid solubility of the 18-beta glycerinic acid is higher than the glycyrrhizin. Got my point? Right. Therefore, if we produce 18-beta glycerinic acid out of glycyrrhizin, then this is your new phytomolecules, and you can develop it into a new. This is the one example, right? We can take a lot of examples also here. Similarly, another example is, you know, all of you, gymnemic acid. Gymnemic acid is present in Gymnema sylvestre plant, right? That is called gurma. Okay, particularly it is actually used as an anti-obesity, right? Anti-diabetic to control the glucose level, right? We use this gymnemic acid, particularly the leaf of the Gymnema sylvestre. But when we convert the gymnemic acid, right, to gymnemigenin, again the same thing happens here. You see, gymnemic acids are glycosidic molecules, but how they are actually converted into aglycons like gymnemigenin. Now, if you see the bioefficacy of gymnemigenin and gymnemic acid, gymnemigenin are much better than gymnemic acid. But if you see the market, right, most of the product they sell in the terms of only gymnemic acid percentage. Till now, you will not find any product which is telling about what is the content of gymnamic. But actually in our gut, right, in our system, actually the gymnamic acid is converted into gymnamic by the help of gut microbiota. We don't know how much gymnamic is produced inside your gut when you take gymnamic acid. We don't know. But the actual effectiveness actually appears, right, once we convert the gymnamic acid to gymnamic, not the gymnamic acid. Because the bioavailability, if you see, gymnamic is having much better bioavailability than the gymnamic acids. But how we can do it in our lab, in our research? That is the main point, right? Which type of microbe we can select, what type of enzyme you can select so that you can convert the gymnamic acids to gymnamic. Another examples I want to tell you about soyabeans. All of you know soyabeans, right? Soyabeans, actually it contain isoflavones. They are mostly glycosidic isoflavones, and some aglycone isoflavones are also present, like genistein, diadgine, glycitin. Okay, these are all soya isoflavones present in much larger quantity in the soybeans. But if we convert into aglycone molecules, they are having much better effectiveness. Particularly the diadgine, okay, and glycitin are much better effect, right? Like you can see the diadgine is again converted into acetyl. That is a much better effect by efficacy than the diadgine. But what happened in our gut? The diadgine actually converted into acetyl. But we don't know how much acetyl we produce in our system when you take soybeans or soybean seeds. How we can convert this soya isoflavone into the final metabolites outside the system is a challenge, right? That means in the gut, it will do the things. But sometimes what happens, the gut microbiota populations vary from person to person. If somebody is having less gut microbiota populations or very rare gut microbiota populations, right? Or there is a difference in the gut microbiota population, then S equal will not produce in that system, right? But if we convert the diadgine to S equal outside the system, then the effectiveness will be much better. Instead of taking the soya isoflavone, we can take the metabolites of the soya isoflavones. Further, it also produces certain molecules like MK7. MK7 means menaquinone 7s. All of you know vitamin K2, right? Vitamin K2 is menaquinones, right? Vitamin K1 is there, K2 is there, K3 is there, right? But actually use the vitamin K2. In the vitamin K2 also, there are different forms of vitamin K2, like menaquinone 4, menaquinone 7, menaquinone 6, menaquinone 9, right? Lot of menaquinones are there, okay, in the vitamin K2. But out of all, menaquinone 7 is the best vitamin K2, right? But how they are produced? They are produced through the biotransformation or through biocatalysis route. When we take the soybeans, when we culture the Bacillus subtilis, right, in presence of soya proteins or soya products, right, it convert into menaquinone 7, which is actually helpful in sweeping out the calcium from the arteries, right, and prevent the atherosclerosis. If somebody is taking vitamin D, right, and calcium, right, what happens? The calcium will deposit in the arteries wall. If you take in a high dose of vitamins, right, regularly, if you take calcium and vitamin D, right, or vitamin D3, what happens? The calcium will be absorbed, right, and it will deposit in the arteries. But if you are taking simultaneously menaquinone 7 or vitamin K2, what will happen? The calcium will sweep out from the arteries, right, and it will target to the bones. And it will prevent the atherosclerosis. That means you can imagine what is the beneficial effect of this menaquinone 7. You can see the formula, right? A lot of calcium-based formula, you will find vitamin D3, calcium, and menaquinones. All three will be there in one formula, one tablet or one capsules. These are the things, right? What we discussed, right? Soya isoflavone, glycosidic form, that is diadgine, genistein, and glycitin. When we do the fermentation or biotransformations, the aglycone molecules are formed like diazine, genistein, and glycetine. Right? Again, this aglycone is converted into S-equal. You can see the S-equal is very important molecule, right? Which can nowadays used by Japanese peoples, right, to prevent bone loss, to increase the bone mineral density. Diazine is out of date, right? Diazine or soy isoflavone is out now, right? And S-equal is entering into the market. And now in India, we will not find any product contain the S-equals. This is a new molecule. Similarly, we can also convert other molecules also, like ferulic acid can be converted into vanillin, right, by using the microbial cells like Bacillus subtilis. We can also convert tannins to gallic acid. All of you know polyphenolic compounds, right? That is very important antioxidant property they are having, polyphenolic compounds, right? And how we can produce gallic acid? Tannins is there, right? In leaf or in bark or any of the materials. But if you are converting the tannins to gallic acid, then it is more useful. But how we can do it? By using enzyme or by using the microbial cells, right? We can convert the tannin to gallic acid and we can use it as a antioxidants. Now again, another important molecule, all of you know ashwagandha, right? In ashwagandha, you will also find vithanosides, right? Now these vithanosides actually convert into pominol. Now this is the active molecules, right? All of you are thinking that vithanosides is active. No, vithanoside is non-active molecule. I can show you later on, right? Because of large molecule, because of poor lipophilic nature, right? The drug likeness property of the vithanoside is 0. But if you convert this vithanoside, right, into pominol, that is aglycone molecules, then the bioactivity enhances. But how it possible? It is possible only in the gut by the help of microbes or gut microbiota. But sometimes the gut microbiota may not present in your system. Then whatever the vithanoside, right, you will take, it will not convert into sominol, right? That's the active molecule. Therefore, we have to do it outside the system. This is the research. This is the work we have to do. Next slide. See, simply you can do the Swiss ADME. You can go through the Swiss ADME and you can see the ADME profile of both the molecules. You can see the boil egg model. Boil egg model. The first one represents the vithanoside, right? And second one is represent the sominol. Now, if the molecule is outside the boil egg, all of you know that the drug likeness property is 0. The bioavailability of the drug is 0. But once it is converted into active molecule, sominol, then you can see the molecule dot, black dot in the boil egg in the white zone, right? That means if it is there in the white zone, then the drug likeness property is increased. It will be more effective. Got my point? Therefore, the transform molecule is very important. Another example, all of you know Aspergillus racemose, Sotavari, right? There is a molecule called Sotaverin 4 again. This Sotaverin 4 again converted into active molecule like saropugenins. Saropugenin is the important drug molecule, right? If you see the Sotaverin 4, right, they are not drug, right? They are actually the glycosidic molecules having poor bioavailability in our system. But once you convert the Sotaverin 4 into saropugenins, then it became active and highly beneficial, right, in PCOS, in PCOS, particularly the sotaverine 4 is used, but not sotaverine 4, it is the sarcopygenins. Right, and also it enhances the bone mineral density. Clear? But how we can do it, that is the important thing, right? Sotaverine 4 to sarcopygenins. You can see here, sotaverine 4, right, and then sarcopygenins. Now you can see the molecule is, the sotaverine 4 is out of boilet. There is no, we will not find any indication of molecule there, right? That means if you see the boilet, if it is there in the white zone, right, that means the bioavailability is more, right? If it is entering into the yellow zone, that means the blood-brain barrier, it crossed the blood-brain barrier. Here, when you convert into sarcopygenins, right, you can see the molecule present in the yellow zone. That means what? There is a demerit also here. Demerit means what? The molecule can pass the blood-brain. If the molecule is targeted to protect the brain, or a neuroprotective molecule if you want to develop, then the molecule must present in the yellow zone. Right? If it is present in the white zone, right, that means the bioavailability is increased. Now you can see the molecule activity, or when there is a transformation, the molecule activity is enhanced. But what is the quantity of sarcopygenin produced when you do the biotransformation? That is important. Right. Now this is the comparisons. Clear? When we do the Swiss modeling, Swiss ADME, you can also use the ADMET 3.0. Only by no need to do any experiment here. By using the softwares, you can see whether the molecules are having drug likeness property or not. But you need the structure of the molecule. Next, how we can proceed the work, right? That means once you have a concept, of convert any research, right? Here we are discussing about the biotransformation and developing new phytomolecules. First, you should have a concept. First, you should have a theoretical concept, right? Then we have to carry out some preliminary work. Okay, whatever the concept you have, right, we have to do some preliminary work in our laboratory. Then once we have finished our preliminary work, right, we have to apply for the grant, right, from the government agency or from private parties, right? Because you need to do the research. For that purpose, you need funds, you need money. How we can do it? Preliminary work you can do in your lab by the help of your guide or by the help of your teacher. But once you finish the preliminary work, you should ask for fund from the industries or from the government agencies. Then you can get the funds, right? And then you can do the rest of the work. Then whatever the fund you will receive from for the grant, right? Then you have to complete the work within the stipulated time period. Then you have to apply for the patents and you have to go for the publications also, right? Sometimes we also do not go for the patent or publications. We keep it trade secrets. That is a much better than patent and publications. Then after that, industry partner and commercialization. Once you have finished the work, right? Once you have a published or having patent or you are having the trade secrets, then you can commercialize the product. You can start your own venture, right? You can carry out your startup industries, right? Or you can transfer the technology to other industries. Or the person who has already funded your research, they can also take your process. This is what actually we do in academics, in universities. But for student, right, This is very important. Once you finish the work, you can also start your own venture, right? That means you can start your own industry, right? To set the molecules, right? For example, if you see in the market, right, what we have given the example, right? Gymnamigenin. Gymnamigenin product you will not find in the market, right? Till now, we don't have any product saying that it is containing this much amount of gymnamigenin. But if we have developed a product containing a high amount of gymnamigenin, that means you have succeeded, got the success, right? You can carry out that product into next level to commercialize. But how we can design the research? How we can start? First, we have to select the herb, right? That means the source for the natural products. What is the source? First, then after that, you have to see the phytochemistry of the molecules. That means whatever the phytomolecules present in that herb, what are the phytochemistry of that, right? Then we have to develop the phytochemical analysis. That means we have to develop a chromatography system. Okay. That means we have to develop the method to analyze the phytomolecules. Because sometimes the molecules may be new one, right? It may not be there in the. Therefore, you have to develop the phytochemical analysis method. That means you have to develop the chromatography, right? Then after that, we have to screen the microbes or cells which can convert the molecule into another molecule. The microbe selection we can do, right, from the gene bank, or we can carry out microbial screening from the gut. Okay, that means you can feed the herbal products or natural product into mammals, right, or in animals, right, and you can collect the fecal matters or in the GIS excreta, right, and then after that you have to collect the microbes which are actually converting the molecule from one to others. And those microbes are actually highly beneficial to convert the or to carry out the biotransformations. But sometimes some microbes are difficult to culture. Whatever the microbe we will isolate, that must be grass category, right, and should not be pathogen, right, and it should very easy to grow, right, and should tolerate the high concentration of the phytomolecules. Then only you can able to transform it to the molecules. Then next, we have to see the enzyme. Which enzyme is responsible for converting the molecules? Because sometimes what happens, if you are doing transformation by using the microbial cell, it will create a metabolite mixture. It is a mixture of different metabolites. But once you identify the enzyme, right, that means you can convert the only single type of metabolites. And you can carry out biocatalysis, not the biotransformation there. Then once you have a molecule in our hand and you have a microbe in your hand, you know the enzyme, right? Then we can carry out the bioprocess development. That means we can do the fermentation. That means what? We will take the herbal extract having known molecules, right? And you can ferment the herbal extract by using your microbes, right? And to convert into other molecules. That means we have to develop the bioprocess, right? Or fermentation technology. After that, whatever the bioprocess you have done, right, then whatever the molecule is formed after the biotransformations, that you have to again purify and you have to check the structure. What type of molecule is formed after the biotransformations? Exactly what we thought, right? What, according to our concept, whether it is produced or it is producing a different molecule. That means after the biotransformation or after the bioprocess, we have to purify and we have to analyze the biotransformed or metabolites. Once it is done, then we can go for next work, that is immobilization of the enzyme. We enhance the biocatalysis process. Then we can also have bioactivity, okay, and also we have bioefficacy studies, right, and also we have this in vivo bioactivity, toxicological profiles, or that molecule, whatever you have developed after the biotransformations. right? This is what actually I already discussed, that is the selection of the herbs. Selection of the, after the selection of the herbs, we should also have a plant extract, what already sir already told in the previous lecture, how we can develop the plant extract, right? And how we can analyze the phytomolecules, okay? Plant extract and molecules, all the three things you have to analyze. Because if your selection is wrong, that means everything will be wrong, right? First, the selection of the herb, and whatever the extract you have prepared, that must be rich in the phytomolecules, right? And whatever the molecule you are analyzing, that must be pure, right? Or it should known to. Otherwise, the metabolite will be a different one, will be confused. Therefore, all the three things are very important: the extract, the phytomolecules, right, and the herb. This is what already discussed, right? The extraction and chemistry, chromatography, and how we can do analysis. Chromatography, mostly we use HPTLC, high performance thin layer chromatography, or we can use HPLC, right? Or we can use LCMS, right? We can use LCMS, TLC. Lot of techniques are nowadays available, right, to characterize the phytomolecules or characterize the extract. See, this is the example, right, of HPTLC profile. You can see in the first chromatogram, you can see the peak at second position, that is unhydrolyzed. That means before biotransformations. But once we transform the phytomolecules, right, you can see the gymnamigenin content is increased because of biotransformation process, because of biocatalysis. These things you have to analyze, okay, during the bioprocess, during the biotransformation process. Otherwise, you will not able to understand whether the biotransformation process is going smoothly or not. The target molecule are formed or not. Therefore, you have to do this analysis. Right? You can go for different type of extraction techniques also to enhance the phytomolecule presence in the extract. You can see, sir already told ultrasonications, right? When you do ultrasonications, what happens to the plant? You can see the electron microscopy. Now, when you do ultrasonications, there is a cavity formed, right, in the plant molecules, right, and the extraction process is enhanced. Okay, and the phytomolecules, whatever the phytomolecules, can be extracted properly when you do the extraction by this ultrasonication process. Now, the screening part appears, right? Screening means the screening of the microbes, right? Screening, then after that, identification of the microbe, because it should not be any pathogen here, right? It should be a grass microbe, right? And then we have to carry out the bioprocess, then bioprocess optimization, because the metabolite sometimes what happens, produce in less quantity. Therefore, you have to optimize the bioprocess. You have to optimize the fermentation process. And then after that, whatever the molecule is formed, that we have to analyze. And then after that, we have to carry out the activity. That means all the 6 things are very important: screening of the microbe, identification of the microbe, bioprocessing the microbe by using the plant extract, right? Then optimization of the bioprocess, then the molecules, whatever it is produced, that you have to analyze, then the bioactivity part. Okay, this is already we have discussed, right? That means we have to, the screening of the microbe we can carry out from the gene bank or from the environment, right? Or from by using the targeted cells also. We can carry out the screening of the microbial cells or biotransformation process. we can do the identification of the cell, right, by genomic and by using the proteomic tools. Genomic tools will give you idea about what type of microbe it is, okay, during the secondary screening process, and proteomic work will give you the enzyme which is responsible for the biotransformation process. Both the things are necessary here. That means we have to use a genomic tool by using different techniques. That is a lot of techniques we use here, right? That means you can use 16S ribosomal RNA sequencing, right, to identify the microbe. And for that purpose, you have to first isolate the DNA. Then after that, you have to carry out the amplification of the target part, right? And then after that, sequencing is done, right? And from the sequencing, from the phylogenetic tree, you can identify the cells. This is what the phylogenetic tree, and from the phylogenetic tree, right, you can identify what type of microbe you have isolated for the biotransformation process. These are all these things we can do by using softwares, right? Online softwares are there, right? You can do phylogenetic trees, right? But isolation of the DNA, sequencing part, right, and isolation of PCR, all the things you can do in the wet labs, not in the software. And this is the proteomic part, right? Whatever the enzyme are produced by the microbe, what type of microbe, what type of enzyme they produce, how actually the microbe convert one molecule to other molecule. Because somebody may ask you that you have taken a microbe, you are converting one molecule to other molecule, but which enzyme is responsible for conversion of the transformation? That we have to also tell. Because if you are not knowing the enzyme, then you cannot do the biocatalysis later on. Biotransformation you can do, but not the biocatalysis. Therefore, we have to identify the proteins also, enzymes. This is important one, right? Gut microbiota. Gut microbiota, how we can generate, how we can modify the gut microbiota in the lab. For example, if you take an animal, right, rat, okay, lab animal, right, and first what we do, we clean the gut microbiota initially by treating with antibiotics. Then what we do, we feed the plant extract, right, for several months to the same animal, right? After 3 to 4 months of treatment, we observed that some type of gut microbiota are generated inside the animal, right, which actually convert the phytomolecule to other molecules. Once you confirm the gut microbiota were generated in the animal, right, then the animal were sacrificed. The microbiota, whatever the microbiome, right, or present in the gut, right, they are collected, and then after that, individually each microbe is used for the screening purpose. This technique we use to develop S equal, right, from the diet gene. What we have done, right, we have taken animals, right, like rat, okay, and then we have feed diet gene to the animals regularly, okay, for 4 months. Then after that, after four months of feeding, right, we observed that one type of cells are overpopulated in the gut of the animals. Those microbes were actually cultured in media outside the gut, right, and they are used to convert the diet gene to a sequel. And sometimes we do metagenomics. That means if the gut microbiota is actually a mix of different type of microbes which are not easily culturable, right, then we have to carry out the metagenomics to understand what type of microbe are present in the gut. And then we analyze the metabolites. What type of metabolite are formed, okay, when you are actually growing the microbes, right, in presence of the starting materials. What type of metabolite are formed? If the target metabolite is present, that means the gut microbiota is also present, which is actually converting the molecule to one to others. Okay, that means the metabolite what is formed, that may be target or that may not be target also, right? And then the metabolite are purified by chromatography techniques like preparative HPLC. Here we have to use a different techniques, preparative chromatography you have to carry out, right? Like counter current chromatography, right? Or we can carry out preparative TLC, right? Or we can also carry out preparative HPLC, right? Or we can also carry out preparative column chromatography. And then after that, we have to identify the metabolites, right? By using the spectroscopy like mass spectroscopy or by using this NMR spectroscopy. Now, once we have done with the metabolite profile, right, and also we have a genomic and proteomic profile, right, then you can club both the things to identify the pathway, how the microbe is actually converting one molecule to other molecules, which protein is involved, which enzyme is involved, which gene is involved, right, and what type of metabolite is found. If we know the whole pathway, how the transformations occur by the microbe, then we have a clear picture. It is not that only the metabolite, right? We have to also tell about which gene is there, which gene is responsible, which protein is responsible for converting the starting material to the metabolites. Then only your work is fulfilled. Otherwise, it will be half. Got my question? Right? That means here also very important, right? Now, whenever you are doing a biotransformation, you have to check also, right, whether they are transformed properly or not by using TLC, HPTLC. Because in HPTLC, what is the benefit here? Simultaneously, you can analyze lot of samples, which is not possible by the help of HPLC. Therefore, simultaneously, if you load lot of samples, you can see which one is best, which one is best transformed. And these things already I discussed, right? Because you have to do the spectroscopy, IR, NMR, mass, okay, everything you have to do to identify the metabolites. Now, this is the bioprocess, right? Once you have done these things, now we have to carry out bioprocess. Bioprocess means the fermentation. Because you are using a microbe here. You can use also mammalian cells here, but mammalian cells are not till now not used for the developing new drug molecules. You can use only the microbial cell, and that is the bioprocess. You can go for the SSF. SSF means solid state fermentation process. Okay, or you can go for the SMF, that is submerged fermentation. That means in a reactor, you can load the microbe, whatever you have developed, right, or isolated or screened, right, and also the extract, plant extract, right, then the process will start, right, and the conversion will start. But once you don't have the extract, right, for example, you have a crude herb, right, or powder of the herb, right, then you can go for the solid state fermentation. That means directly you can load the microbe, right, in the herb, right, and the fermentation will start, bioprocess will start, and the metabolite will start producing. Both the way you can do. You can carry out solid state fermentation, right, or if you have extract, then you can go for the submerged fermentation to develop the molecules or to produce the molecules. Now, once the process is done, right, the yield may be very less. Yield optimization is very important. That means that whatever the product is produced, right, we have to optimize. How we can do optimization? We can go for any statistical techniques, right? Like you can use RSF, surface response surface methodology using box Venn design, central composite design, full factorial, half factorial, right? Lot of designs are nowadays available. You can use design expert software, right? And you can optimize the process to increase the yield of the biotransformable molecule. Clear? And also you have to see the kinetic of the process. Kinetic means what? at what time the metabolite are formed and how much quantity of a metabolite are formed, how much substrate are reduced, okay, how they are actually converted into the molecules. The kinetic, fermentation kinetic also you have to study. That means pH, what time, right, they are actually changed. Then only you will get the optimum product. Once the bioprocess is over, now we have to switch over the enzyme, because the microbe sometimes may produce non-targeted metabolites because of biotransformation. But if you are using the enzyme, then it will create only one type of metabolite. Right? Therefore, we have to understand what type of enzyme actually responsible for converting the molecules. Once you identify the enzyme, then we can go for the separation of the enzyme, right? Then purification and characterization of the enzyme, and same enzyme can be immobilized. Can be immobilized, right? And you can use this immobilized enzyme, right, for converting the molecules. We have used a lot of fibers, right, to immobilize the enzymes, natural fibers. You can see here the fiber. Okay, they are actually, this is the electron microscopy picture of this fibers, right? You can see a lot of pores, right, in the original fiber. But once you immobilize, they are full packed, right, with the enzymes. And these things can be used for converting the molecules. Once you have a molecule in your hand, then we have to go for the biological efficacy study, toxicological profiling, ADME studies, lot of things you can do, right? So that you can create a comparison, right? Once you have biological evaluation, right, then you can compare the biological efficacy of the original molecule and the transformed. Then only you can sell your product. Otherwise, nobody will buy your. If you have clear understanding that this molecule is produced and having much better bioactivity than the original molecule, okay, then only you can able to sell the product in the market. Everybody will look at your molecules. Yes, selection, screening, and activity. These are the processes. Summary of, you have to plan, then you have to carry out discovery, right? Discovery means the microbes, cell, enzyme, right? Which microbe, right? That discovery part. Then evaluation. That means bioactivity, bioefficacy, pharmacokinetic. Okay, these things, evaluation. Then after that, you can go for the patenting, right? Then you can go for the manufacturing of the molecule, right? Then you can able to market. Then again, the process start. Once you market, that means you will get the funds, money, right? Then again, you can plan new concept, right? And you can again same work you can do. Otherwise, if you stop at some point, that means it is not the final work. For example, you have done the discovery, but you have not done the evaluation, then stop. You have done the patenting, but you are not able to manufacture it, then there also stop. If you are able to manufacture, but you are not able to sell it, then also it will stop. Therefore, all the process is very important, right, to create the molecule and to again carry out the research further, next level. Then only you can continue the work. With this, I like to thank of all of you, right, for the program. Any questions you can ask?
Thank you, sir, for this wonderful presentation of yours. I must say it was really a thought-provoking presentation, and it really provided a huge lot of will to our minds and thoughts. So, if we have any queries, firstly from the audience over here, I request the student volunteers to please carry forward the mic. Any queries from the audience? Yes, we have one query.
Just two or three molecules that is not in marketed formulation in India. So what is the possible reason? Have these molecules never came under the kind of pharmaceutical company? Pharmaceutical companies actually till now not able to develop the process that to create this molecule. They know this molecule is better than this, but they don't know the process how to produce this molecule. This is the thing that we have to do. Everybody, no, in foreign country also, they actually now new concept. Now all of you know that the postbiotic. All of you, I think all of you know postbiotic, probiotic. I think all of you know. Prebiotic, you know. Symbiotic, you know. Postbiotic, any of you know? Postbiotic. Postbiotic means what happens? Right, when you ferment the herbs, whatever the molecule produce, right, they called it postbiotic product. The concept is there, but we don't know which microbe is actually converting the molecule. We know this molecule is better than this, but how we can get that molecule? But if we have clear understanding, but to produce it, to you know the process, then we can develop. This is the things. Okay, thank you.
Do we have any other questions? Do we have any queries from faculties as well? If you have any questions from faculties. Sir, I have a question for you. You have already showed one of your slides regarding gut microbiome. Right now, we are also working, one of my PG students saying, so we are now planning for identifying spatial gut microbiota being altered with a selective phytoconstituent. So what would you suggest that what are the spatial markers that we must go for to enhance our publication of results?
Okay, in two ways we can select the microbes, right? Number one, the screening of the microbes. Screening of the microbes or gut microbiota, whatever the profile you have changed, it may be culturable or it may not be culturable, right? If it is non-culturable, then you have to go for the metagenomics, right? If the microbes are culturable, then you have to culture in the media. Like MRS media you can use, right? LB media you can use, nutrient agar media you can use. You can culture the microbes. But sometimes the microbe may not be cultured. Time you can go for the metagenomics to understand what type of gut microbiota are produced because of feeding. Now, second option is that metabolite screening. That means once the microbe is known or unknown, right? You have to screen the metabolite. What type of metabolite are produced when you feed the plant or plant extract or molecules? If the metabolite are produced in the excreta, right? That means what? The microbes are present, okay, in the gut and they are actually converting. You can check the urine, right? You can check the plasma, right? You can check the fecal matters, right? And see how much metabolite are produced, okay? And what type of metabolite they are producing. This is the two way.
Sir, do we have any gene bank or something that
No.
enable us to know based on the nature of like if it is a terpenoid molecule, what type of microbiome it is going to generate or something?
No, not.
So we have to go
You
for
have to screen. You have to screen because these are all new one, right? We don't have database. We don't have any prior knowledge. What happened right, in the gut. What is happening inside the gut? Because the microbiota or populations change accordingly. Actually, in our lab, right, we train the microbes. How we do? Right, actually we feed the microbes regularly with a one molecule for years and years together. Almost all 8 to 9 years, we feed a particular molecule to a microbe. Then what we have observed that after feeding for several years, right, the microbe actually change its genome. Okay, that means slowly and slowly they change their property to convert the molecule to other molecules. Initially, they don't have that genome in their system, but once we feed a molecule to them, right, regularly at a slow level, that means at lower concentration, right, then we have observed that after several years, they change their properties, their genomic property change, and the metabolite are produced. Particularly in, I think all of you know, endophytes. Endophytes, lot of endophytes are there in the plants. Actually, they produce the same metabolite which is produced in the plants. How they produce? Because they adapt to the molecules, to new environment. But once you remove the microbe from the plant, right, and you will culture it for 6th generation, right, on the 7th generation, you find the microbe will lose its character to transform the molecules or to produce the molecule. They actually adopt, they change their genomic character. Therefore, we don't have any prior gene set, right, which is actually converting, or we have to do.
Thank you, sir. I personally look forward applying the insights shared by you in my research work. And most of us present over here were really, it was a gaining experience for all of us. I would now request our Professor Dr. Anuradha Mishra ma'am, Director AIP, and Professor Dr. S.P. Singh sir, to please join us on stage to felicitate our honorable speaker with a memento. I would also request Professor Dr. Narahari Palekar to be here with us on stage for the felicitation process. Thank you, respected dignitaries. Thank you, sir, once again. Respected dignitaries and dear delegates, the speaker of the last technical session of Day 2 RBPS 2025 is Dr. Deepak Chitkara, Associate Professor, Department of Pharmacy, BITS Pilani. He is going to join us on the online platform, either the Zoom meeting. May I now request our IT help to please visualize the on screen. Thank you, Aman sir. May I now take this honor of officially introducing Dr. Deepak Chitkara, the honorable speaker of the last technical session of Day 2 RBPS 2025. He did his PhD in pharmaceutical sciences and MS Pharm in pharmaceutics from National Institute of Pharmaceutical Education and Research, SAS Nagar, Mohali, Punjab. Presently, he has been working as an associate professor at BITS Institute, Pilani. Our speaker has also worked as a postdoctoral research associate at the UNMC OMA NE USA and as a visiting research scholar at UTHSC Memphis TN USA. He has published over 65 research articles till date and 2 book chapters and almost 6 patents till date. He received the prestigious Grand Maxi Science Scholar Award in the year 2011. We are blessed enough to have you, sir. Thank you for joining online. May I now request you to kindly save your slides and proceed over with your presentation.
Yeah, so thank you for the nice introduction. I hope I'm audible
Yes.
to everyone present over there,
Yes, sir, you are.
and my slides are also visible, right?
Yes, sir.
Okay, so thank you. And first of all, I would like to congratulate the Amity Institute of Pharmacy Lucknow campus for organizing this unique conference. But and the topic is quite interesting when we see through it that recent breakthroughs in pharmaceutical sciences. And one thing that intrigued me is that herein we are talking about taking the innovations from bench to laboratory and also going beyond. And one thing which my previous speaker, Professor Vivash Bibhu Prasad Pandaji, have already highlighted on the roadmap. When we look at the roadmap, that how we move from the entire journey which a research takes, and ultimately we are looking that it should be translated into some form. So that is what I try to correlate in my session as well, and particularly focusing on three components. So as you can see in this particular title, we have precision therapeutics, we have nanotechnology, and the third one is CRISPR-Cas system, which is a tool that can do the gene editing. We can identify a particular gene and can do the editing very precisely. Now, how the precision medicine or the precision therapeutics could be achieved by utilizing this tool and utilizing the power of nanotechnology, and try to bring forward some of the research data that we have obtained in the recent past. So currently I'm at BITS Pilani campus, and also had an opportunity to look on the other side of entrepreneurship, where I founded one company known by the name Nanograde Innovations Private Limited that works at the junction of the nanotechnology and how we can utilize these nanosystems to take it to the market. So moving forward, so this is what BIT Pilani campus is. So currently there are 5 campuses: Pilani, Goa, Hyderabad, Mumbai, Dubai. And the Pilani campus is around 328 acres. And right now I'm placed somewhere over here, sitting in my cabin. And BITS Pilani has a long legacy. It was first started in 1901 as a school. Then later on, it was converted into an intermediate college in 1929. Then this pharmacy college, or pharmacy, what we have right now, the Department of Pharmacy, it was first established as a Birla College of Pharmacy in 1950. And currently we are celebrating our 75 years of existing, and again looking forward to move forward and have do collaborations with many other institutes. Now coming back on to what the lab is doing. Now, when we look at different molecules that can be used for therapeutic purpose, we can have the small molecules, we can have the macromolecules. The small molecules wherein we can identify a particular target that the small molecule can go and bind to a particular receptor. And then there is an array of downstream signaling that occurs. So in order to cure a particular disease, but there is also a class of molecules which we put together as the macromolecules. And in that we have proteins, peptides, the oligonucleotides, genes. So can we utilize them also for achieving a precision medicine? Now, if you look at the definition of the precision medicine, as per the US FDA, if you look at over here, precision medicine, it is also sometimes we can call it as a personalized medicine, is an innovative approach of tailoring the disease prevention and treatment that takes into account the differences in the people's gene, environment, and lifestyle. So what it also brings in, that can we have the right treatment for the right patient and at the right time? So that's what my lab is also looking after, that can we identify a particular gene which is involved in the disease progression, and using these macromolecules, be it the genetic materials such as microRNAs or plasmids, or certain proteins such as CRISPR-Cas system, can we overcome or alleviate the disease condition? So we all are aware of the central dogma of molecular biology, where we know that DNA makes the RNA and RNA makes a protein. So we have the DNA, which is present inside the nucleus. Transcription happens, that forms your messenger RNA. That messenger RNA is exported out, and ultimately it is associated with the ribosomes, forming the protein. And these protein molecules are then later on involved in various other processes. Now, in a disease condition, when we say we have a patient of a cancer or any disease you just pick, you can identify that the reason either the protein had got mutated or it is not doing its job, or the protein which is necessary is not being formed. Then we go back slightly and look at the mRNA, whether that mRNA is there, whether it is upregulated or downregulated. And that is again related to the gene that is transcribing it. So there is an ample of opportunity to work either modifying the gene by doing the genome editing. We can knock down the gene or we can correct the mutated gene and then influence the entire downstream pathway of making that RNA and then the protein. Another strategy is doing the RNA interference. So for example, the mRNA is overexpressed for a particular protein and we want to bring it down. There is a way by which we can let this mRNA to bind with some other molecule like a siRNA or a microRNA and influence its translation. We don't want that the translation to happen, so that we can correct the disease. So that comes under the RNA interference, where other RNA molecules, they are inhibited, the mRNA is inhibited, and ultimately there is a downregulation that had happened. Now, my lab is basically working in these particular domains where we are using this nanotechnology approach, such as having different biodegradable polymers. Particularly, we look after polycarbonates and then combining them with various lipidic systems as well to design these nanosystems and utilizing them for delivering these either the gene editing tools or the mRNA therapeutics or the microRNA, so that ultimately we can have the precision medicine done. Now, going back, that how it all started. So if you look at the history of gene therapy, so the first, this double-stranded DNA, as we all are aware of, has been discovered somewhere around 1950s, where the structure of the double-stranded DNA is there. And then the concept of this gene therapy, that we can influence the DNA and further downstream steps could be also modified. That was conceptualized somewhere in 1970s. That led to the first gene therapy trial that happens in around 1990. So there is a huge gap that is happening, because that's how the research moves. That requires a lot of thorough experimentation and then finally analyzing those data before it actually reached for the clinical practice. But the initial trials that had happened, there were certain failures as well. And if you just consider this entire timeline, it is somewhere around 2018, the first gene therapy based formulation, particularly utilizing the non-viral vector, has reached the market, which got the FDA approval, which is on Petro. And after that, there are many other products. Well, we have around 6 products which are based upon these similar to these on Petro, which are based upon siRNA, and utilizing these non-viral vectors have been developed, such as givosiran, lumisiran, inclisiran, natosiran, butisiran. So they basically target a particular pathway and can influence the further translation process. Now, at the same time, the gene editing could also be done. It could be utilized. So there is another set of experimentation, another direction that is also there that I'll just touch at a later stage. So if you look at the RNA interference, so it's not only the siRNA or microRNA that could be utilized for doing these translation, blocking this translation or doing the RNA interference. There are several other approaches as well. For example, using antisense oligonucleotides where a single-stranded DNA is used, that can go and bind to the messenger RNA and influence the translation process. Then there are certain triplex forming oligonucleotides, aptamers, they have also been used. Then several other strategies using ribozymes, DNA enzymes, then siRNA, microRNA. Among these, also, if you see, there are a lot of publications that has been done in the area of siRNA, which are around 20 to 25 base pair RNA oligonucleotides and can bind to the mRNA and cause the translation repression. The mRNA get degraded, so it is not converted into a protein. On the other side, we also have microRNAs. And these microRNAs are also 20 to 25 base pair sequences that can bind to the target mRNA. Now, these two basically differ in their sequence specificity and how other, but both of them have the same target, which is the mRNA, and ultimately leading to the translation repression. So we started focusing into the delivery of these messenger RNAs that delivering these microRNAs that can utilize them to do the personalized medicine and also to combat some of the diseases where these small molecules may have challenges, may face issues in terms of resistance. So the first step, when we say we are looking at the microRNAs, so microRNAs are also present inside the cell and they play a role. Now, if you look in a cancer cell, so there are several microRNAs which are either upregulated or downregulated. Now, they are known to control certain functions inside the cell by controlling the mRNA. Now, in a disease condition, what had happened that their levels have been they have the aberrant expression. Like we have this LET7 microRNA or microRNA 31, which is involved in migration and invasion. On the other side, if you look from epithelial to mesenchymal transition, there are several microRNAs that have been reported to impact to inhibit this EMT transition. Likewise, there are microRNAs which are involved in chemoresistance, particularly pertaining to the cancer stem cells, such as microRNA 200 family, LET7 family, microRNA 205. So these microRNAs are indigenous, they are already present in the cell and they are playing a role. Now, can we utilize these microRNAs? Let's say if there is a microRNA which is downregulated in the cell and that is ultimately causing a disease. We are seeing the manifestation of that disease. Now, since these are 20 to 25 oligonucleotide sequences, so is it possible to deliver these sequences to the cell? Yes, of course. Theoretically, we can say that we can utilize a microRNA mimic and deliver it. And if there is a microRNA which is overexpressed inside a cell, we can inhibit that microRNA by using an antimer of that. So there is a possibility of using these microRNA mimics or antimers, and we can control the translation of the messenger RNA. So the first question that is asked, that how to identify from where to get the data that which microRNA is overexpressed, which microRNA is underexpressed. So profilings would be done for that purpose. Now, when we look, so we can take an example of a cancer right now. Now, identifying these microRNAs and their targets could be done in 3 different levels. One is that using the in silico modeling, we can identify the target genes which a microRNA can have. So there are several tools which are available online, freely available, and that could be used to do this in silico modeling and can identify that this particular gene or a protein which is expressed from a gene is involved in cancer, be it EMT, be it in cancer stem cells. Now that protein or the messenger RNA which is coding for that protein could be identified, and the microRNAs which can target that messenger RNA could be identified using in silico modeling. Another way is doing it in the cell cultures. We have a number of cell lines and also the cell lines which are resistant. And from these cell lines, the cancer stem cells could also be isolated. So procedures are already there, protocols are set. So we can isolate these and identify the expression of these microRNAs and compare with the normal cells. And if we are having the resistant cells, we can compare it with the non-resistant cells and identify that which microRNAs are playing a role. And another strategy is using the cancer tissues. Now, when we can take a pancreatic cancer tissue, extract the microRNA, and these tissues could be further grown in animal models by serially implanting them so that we can have a sufficient amount of that tissue and identify the microRNAs by doing the microRNA profiling in them. Now, what we did, we started with identifying these microRNAs somewhere in year 2011 or so, wherein we started looking in the pancreatic cancer. So what had been seen that in the case of cancer stem cells, as well as in case of certain patient samples, there are set of microRNAs which are either overexpressed or underexpressed. And this gives a cue that there is a possibility of utilizing the microRNA. Now, what we have identified that we can have this microRNA 205, which has consistently shown in case of patient samples as well as in case of cancer stem cells that this particular microRNA is downregulated. So just to set the proof of concept, can we deliver the mimic for this microRNA 205 so that the resistance could be elevated? So when we set up an experiment, we need to have the proper controls and in case of microRNA, like in case of small molecules, we can use another drug molecule which is very well established in the clinical practice and compare it with that. But what to do with the microRNA? So for that, scrambled microRNAs are available that could be used that does not have any target mRNA. So they are used as control. Now, since we were looking in the chemosensitivity or the resistance in pancreatic cancer, so the standard therapy for pancreatic cancer, which is using this gemcitabine. So combining this microRNA with gemcitabine has indeed, we were able to see the difference that the sensitivity is improved. So here the cell line that is used is the chemo-resistant cell line, which is made resistant towards gemcitabine. But that gives enough proof that definitely these microRNAs could be utilized for targeting the resistance. But the real challenge is on the delivery, because we have a molecule, oligonucleotide, 20 to 25 base pair long, highly hydrophilic in nature, prone to degradation, easily degraded by RNases. So how to give that? So there comes the role of nanotechnology, because when we talk about the translation of any product which we are doing in the research and we want it to see the light, so there are several key questions that we ask, such as whether the work which we are doing, does it has a market potential? And you can ask various companies, discuss with them, and try to identify that do really there is a market which is existent. Because if you make a product and cannot sell it, there is no market, then you can't do anything about it. Another thing is the clinical need, which falls in our domain as a scientist. We look for whether there is a requirement for that. There is a clear unmet medical need which is present. And whenever we try to identify that clinical need, there are three things that we can look into. First is the safety, another one is the efficacy, third one is the compliance. Safety, when we say that the new product that we are designing, it is safer than the existing product. We are able to avoid some of the toxicities which are associated with the already products which are being used, the conventional therapies which are being used. Next is the efficacy. Are we able to improve the efficacy of the product compared to the one that we are having previously? So that is another part. And the third one is the compliance. So in that case, the patient compliance could be improved. Any advantage to the patient side, are we able to get that? That's what we look into. So these all revolves around the physicochemical properties of the drug. We can identify a challenge with them and try to solve that challenge. It revolves around the toxicological properties of the molecule. If you are looking at the safety profile or the biopharmaceutical properties, looking into the ADME of this, how it is getting absorbed, what half-life it is having. So based upon that, we try to identify. Now, if we specifically look into these macromolecules, they do have a challenge. That is why they were not efficacious. If we just give them in the naked form, they are not going to show any impact. So definitely there is a need to develop them, but there are challenges also associated with them. So the next question that comes in, can nanotechnology solve these challenges? And of course, there's have been ample of literature that favors this hypothesis that nanotechnology indeed can deliver these hard to deliver molecules. So looking particularly into these type of molecules such as microRNAs, several nanotech based products are already there in the literature, such as using these lipidic systems, forming lipoplexes, or making these nanoparticles, or forming the micelle plexes with them, or conjugating them, or doing this backbone modification to improve their stability. So these are several strategies which are already reported where the POC has been generated. But we tried to look into from a system per se, wherein we designed a polymer that can deliver these molecules efficiently. Now, looking at the structure of these microRNAs, so they are having the negative charge on them, anionic in nature, and hydrophilic. So one strategy that could be utilized is doing the complexation. So they can complex with a positively charged polymer. So we designed this polymer having a PEG chain that can provide the stealth effect, and a polycarbonate chain. And that polycarbonate chain is further modified so that it can be converted into a nanosystem and has a positive charge coming because of the cationic chain which is present, and then can complex. So an array of experiments were performed. And of course, when we are talking about the resistant cell lines, we need to add the therapeutic molecule, which is the first line treatment, gemcitabine, into it. So the results were also very promising when we look into, particularly in the in vivo studies, wherein we found that indeed, when these formulations were given, they showed the enhanced efficacy as compared to the controls which were used, like giving the free drug molecule or combining the drug molecule with the negative control, which is a scrambled microRNA. And the research was quite promising. Now, next is, if we look into the system per se, is the system is ready to go ahead? That is the next question that we ask, because there are always the next step that we can look into. Because it's not only that delivering that molecule, it has to reach the right target, though there is some proof we have already established. But moving forward, we need to further understand and improve the system in terms of controlling its intracellular trafficking. Now, when we say intracellular trafficking, and initially we have seen that there is a challenge in terms of the structure of that molecule, the microRNAs, which are around 20 to 25 nucleotides, hydrophilic in nature, how they are going to be taken up by the cell. And once they are taken up by the cell, they have to come out from the endosomes and interact with the ribosomes, get associated into the RISC complex, which is RNA-induced silencing complex. So the literature also favors this thing that we can modify on the system. Now, when we are looking into the translation process, now these formulations could be given either the intratumorally, or it could be given intravenously. And once it is injected in the IV, it has to pass through various barriers, like the barriers from the phagocytosis. RES system can take it up. It has to further then extravasate, enter the cell, and once it is entered, either by receptor-mediated endocytosis or by adsorptive endocytosis, it ends up in the endosomes. And it has to come out of the endosomes, so finally to get associated into the RISC complex. So how we can control this intracellular trafficking so that it could reach the target site? So there have been literature that also comes along when we were working on this project that showed that if we can use cholesterol in our system, it can definitely improve its cellular uptake and has influence on the intracellular trafficking. So what is the cellular uptake that we wanted to improve? And the another thing is that it should come out of the endosome so that it is now released in the cytoplasm. So for that, there is some literature that come up that if we use a morpholino group in the polymer that do influence the endosomal escape. So this kind of a group is helping in a proton sponge type of a effect, so that the endosomes were broken down and the complexes are released in the cytoplasm. So then we try to look into that is there a possibility of modifying these systems, the polymers that we have designed. And of course, that we did by modifying the structure, where we retained the cationic chain and the basic structure of that polymer, but added cholesterol and morpholino into it and analyzed that do these systems are able to deliver now. So wherein we were successfully able to deliver these microRNA 34A and then along with another anti-cancer molecule, which is docetaxel for breast cancer therapy, where indeed we saw an improvement, where this particular system can form a nano-sized particles, we call them as nanoplexes, and can deliver inside the cell and are quite efficacious as well. So having learned about the delivery system, optimize this delivery system, the question comes to us that what next in the personalized therapy? Because these systems are already now have been taken forward for further testing. Now the next thing that comes is on the further going down into the central dogma, because earlier we have seen into the RNA interference. Now, is it possible to do the editing and modify the gene if there is some mutation that had happened? Or can we control the expression of a protein by knocking down the expression of the gene? So during that time, when we were starting this work, the CRISPR-Cas system came up, which has been shown as one of the breakthrough. It is considered as a breakthrough in the field of biotechnology. So wherein this particular system has a Cas protein with a guide RNA. And this protein this ribonucleoprotein complex can go and bind to the specific target site and cause the DNA to cleave. So the Nobel Prize for this technology was given to Doudna and Carpentier in the year 2020. Now this particular system was basically taken from the bacterial defense system, the acquired immunity that the bacteria is having against the bacteriophages, and then adopted for gene editing in the mammalian cells. So now you can see over here that we have a CRISPR system bind to the DNA segment. So how this system work? How this CRISPR-Cas works? So there is a Cas protein and a guide RNA. So one sequence of this guide RNA binds to the Cas protein to form a ribonucleoprotein complex, while there is another sequence that is a target specific sequence and go and bind to the specific DNA segment. So once it do this Cas protein, which is a nuclease, it nicks the DNA. Now the repair mechanisms and takes in, which is either by non-homologous end joining or homology directed repair, that lead to the insertion and the deletion in the genes if it is going through NHEJ, or simultaneously we can also provide a donor template if you want to do the correction in the mutated gene. So these are considered as the molecular scissors and gene editing could be achieved. Now you can see in this particular video on how this target specificity is there. This was published in Nature Communications few years back, wherein in the first video, you can see that it is having a target specificity. Okay, so this video is somehow not playing on this. Anyhow, I'll explain that. So we have this Cas protein that is binding to the specific DNA segment, and the same DNA, the same ribonucleoprotein can cleave the DNA after in specific time period that it can cleave, and you can see that the DNA chains, they are going apart. So moving ahead, so is this technology could be utilized for the gene editing purpose? So we know that the first study that came up in 2013, and it has progressed to taking these ahead for the clinical application. And in around 2020, a report came in Nature where it shows that CRISPR gene editing proves safe in a clinical trial in cancer patients. They have very well tolerated them. And that again gave a good boost to the CRISPR technology to be taken forward. Now, when we talk about the entire technology, so there are different formats by which it could be utilized, because we want the Cas protein and the guide RNA to go inside the cell and then to the nucleus and do the gene editing part. So we can deliver them either utilizing a plasmid approach, and this plasmid has a sequence that can code, that can give the Cas protein as well as the guide RNA. So once it is inside the cell, it is transcribed, translated, and then transcribed into the protein and the guide RNA. Or we can deliver directly the mRNA, a Cas9 mRNA, and the guide RNA, which can go inside the cell, assemble to form the ribonucleoprotein. And thirdly, utilizing these ribonucleoproteins itself, assembled outside the cell, and then these could also be delivered. So all these strategies has their own advantages and disadvantages, pros and cons associated with them. Like the third one, it seems to be pretty straightforward and much more applicable because you are directly utilizing the Cas protein to go into the cell and do its job. While in case of plasmid, it has to go inside and then transcription and translation happens, followed by assembly, and then again translocation into the nucleus to show its action. So both has their own pros and cons. In case if we look at the ribonucleoproteins, being a protein and the oligonucleotide present together, is quite challenging to handle and then to deliver. The cost involved is also quite high and time consuming. So each of these strategies has their own positives and negatives associated with them. So having identified that we can utilize these systems for precise gene editing, so we thought what to pick first, from where to start. So then we looked at various diseases, and what we have noticed that one condition is there known as the wet age-related macular degeneration, wherein which is basically a neurodegenerative disease and has been shown is happening because of the overexpression of VEGF-A gene, vascular endothelial growth factor. And it leads to the central vision loss, as you can see in this image. So there is a possibility of knocking down this gene, decreasing its expression levels, so that the disease could be cured. But how to deliver? That's the next question that comes in. So we can have the intravitreal injections, which are done, and then these nanoplexes can go to the RPE layer and do the gene editing part in that using this CRISPR-Cas gene editing tool. So that was the whole hypothesis which was first created, and then we moved forward into looking into its efficacy. But going back, we need a system that can carry these ribonucleoproteins, because these are much larger molecule. We have a large protein, and alongside we have a guide RNA that is forming a complex, which is unstable, and it also has an anionic charge, negative charge. So we thought, why not to use a system that we have already developed for these microRNAs and see whether it can show any impact. And when we started working on these systems, we found that these nanoplexes, which are being formed from the polymer, they were having a positive zeta potential, around 15 to 20 on the positive side. And they can easily complex with these ribonucleoproteins, though a lot of optimization has to be done to get a stable system. And they usually give a size close to 100 nanometers, the blank one. And when we are using these plasmids, because the first approach that we started is using the plasmids that express the CRISPR system. So we were getting around a size of 140 or so. And they were in the nanometric size, with the complexation efficiency, all were looking pretty good. So we moved ahead with doing a set of assays in terms of their stability, in terms of their uptake inside the cells. And what we have found that they were indeed, this system was able to take these plasmids inside the cell, as you can see with the green color, which is coming from the expression of this GFP protein from these plasmids, because these plasmids express the turbo GFP gene. So after establishing all the assays, we went ahead for looking their editing efficiency. So indeed, these were able to cut the DNA, because this Cas system can do that. And in vivo also, after giving the injections, they were able, because we wanted to test to which organs they are going more, because we have two aims over here. One is their in vivo stability that needs to be maintained, plus in which regions, in which organs they are going into. And also, when we are further moving ahead to target the wet AMD disease, how they are going to translocate into the posterior segment of the eye. So these results were also quite promising, showed us that these systems were indeed stable when they were also given in vivo by doing various imaging assays. So after having tested these plasmids, we move ahead for these ribonucleoproteins. Now, ribonucleoproteins being anionic in charge can also be delivered, but the size is quite large with them. So we utilized this strategy of using the lipopolymers that have been designed to make these ribonucleoprotein complexes, which we call it as in nanoplexes of RNPs, and did an array of assays alongside, so that we can identify what size they are getting, which is close to 100 nanometers. They were spherical in shape. When we look in the TEM images, they were able to first complex, and then they were also able to release the ribonucleoproteins, because it's not that the complexation had happened. That complexes needs to be broken down also once they are inside the cell. And for that, a standard assay that has been used is a heparin combination assay that showed that indeed they were able to be released. Plus, they need to be stable. So stability assays in term with FBS, fetal bovine serum is done. That also indicated the same thing on the stability side. Moving ahead on the cytocompatibility, that they need not to be toxic. We want them to be cytocompatible. And even at much higher concentrations, the polymers that we have used and these complexes, they were non-toxic to these cells. That gives us enough fuel to move forward into our cell culture assessments, because now it's a large protein along with the guide RNA that needs to be delivered inside the cell. So we had these proteins which are GFP expressing, so that we can visualize them using confocal microscopy, wherein we observed that within 6 hours, we were able to see the green fluorescence inside the cell, that it is indeed being taken up by the cells and tested both quantitatively and qualitatively using the confocal imaging and flow cytometry assay, where we can see that close to 80% of the nanoplexes are going, close to 80% of cells are expressing this, are having this green color. So that gives us enough idea that indeed their uptake is happening and it is going inside the cell. But the challenge remains, because in order to for this Cas protein to show its action, it has to go to the nucleus and bind to the target site. Because in the previous case, when we were having microRNAs, in case of microRNAs, the target lies in the cytoplasm. But here in the Cas protein, along with guide RNA, has to go to the nucleus. So to test that whether it is going inside the nucleus, there is an assay known as a Cache-Fish assay, which is used. So here in the guide RNA, that targets the telomere regions in the DNA could be utilized. So the guide RNA is designed that can target the telomere regions, and the protein which is used, because we now don't want that it should nick the DNA, because we want to visualize that and do the imaging of it. So the protein that is used is a dead Cas that also has fused with the GFP protein. So this way we can identify that the protein is still there and whether it is going to the telomere region in the cell. And what we have observed that in 48 hours, we can see that inside the nucleus, we have these green dots, which indicates that it is indeed going inside the cell and is binding to its site. So the next proof of concept comes to us that definitely it can go inside the cell, cellular uptake can happen. And secondly, it can also go inside the nucleus and bind to the target site. Now, the real proof of concept is from the editing, whether it can do the editing part or not, where a set of experiments have been performed. One is the T7 endonuclease assay, where we can observe that it is cutting the DNA site, which is with respect to the target guide RNA that we have used. And using Sanger sequencing, also we were able to identify the indels, which it is doing quite efficiently. So all these assays give us a lot of clue that definitely the nanoplexes were able to complex with the ribonucleoproteins, and they were able to go inside the cell, cellular uptake can happen, go inside the nucleus, bind to it, and also can do the gene editing part. So how to move forward now? Because we have these cationic systems, lipopolymeric systems, and they have to, they were showing pretty good data on the editing part. Now, while having discussions and while looking for their application for wet AMD, we thought why not to make them more targeted, because till now we were working on a system which does not have any target ligand attached onto the surface. So next question that comes to us on the targeting side, that if we can make them targeting, we can increase further their specificity. So cyclic RGD is a peptide that has been widely reported to target the integrin receptors. And these receptors, what we have also observed in the literature, that they are also expressed on the RPE cells. So we thought, why not to formulate our system and attach the cyclic RGD onto the system. So a system is further modified with having the cyclic RGD onto its surface, and using the Cas proteins and the ribonuclear proteins that we had. The formulations were made. The size were close to similar to what we have seen earlier, though there is a slight increase, then that could be seen because of the modification in the system that we have done. But they were close to each other. Now looking into how they can go inside the eye, because we have to give the injection intravitreally, and it has to translocate into the RPE layer. So we need to have, right now, it has to cross the vitreous humor, and after crossing the vitreous humor, it has to go to the RP layer. So there should be a diffusibility associated with them. So a simple chick eye based assay was performed, where the chick eye was taken and it was cut, the eyeball was cut circumferentially, and these nanoplexes, that the cyclic RGD1 and the non-targeted one, they were taken and injected into the vitreous humor, and imaging was done with respect to their diffusibility on a confocal. So though the video is not playing over here, but it can show that these dots which are shown over here, that shows the ribonuclear proteins which are present and their movement, and that movement were captured in terms of their diffusibility. What we have noticed that by optimizing the charge on the surface of these nanoplexes, we were able to control their movement. If we have a positive charge which is close to 10, the movement is quite less, the diffusibility is less, which is retarded in the vitreous. But if the charge is reduced, the zeta potential is reduced, they show a much better diffusibility. So we now have a system that could be taken forward for animal experimentation, so that we can test. Otherwise, what will happen, that if we do the intravitreous injection, it will stay over there and will not translocate into the RPE layer. So that gives us further idea that shall we go ahead with the animal experimentation to see that indeed the gene editing is happening. So VEGF-A gene is the target that we have right now for the wet AMD disease, where Vista rats were taken and they were given intravitreal injections, and the editing efficiency. Of course, various other assays were performed for their their ocular distribution, at which location which was reaching. But because of the paucity of time, I'm just bringing in the data for the editing efficiency, wherein in the animals also we were able to see that the genes were cut with an indel efficiency, in indel frequency of close to 10%, by using T7 endonuclease assay or by Sanger sequencing. And definitely we were able to achieve this editing efficiency of VEGF-A gene in the Vista rats using these nanoplexes. Now, is it the end, or it is just the beginning? So we look it as the beginning because it opens up a lot of avenues, not only in case of editing in case of wet AMD, but in other diseases as well, where we can identify that a particular gene which is either overexpressed or aberrantly expressed, can we knock it down? So we extended our research into other diseases, wet AMD, where we have seen we have explored quite well. Then we also looked into in psoriasis, whether we can achieve the editing in the psoriasis condition, because there also VEGF-A is having a role, and in the corneal dystrophies. But for the corneal dystrophies, the delivery system has to be changed. we have these nanoplexes and then incorporated into the microneedles so that they could be applied onto the cornea, and we can see the editing efficiency. So this is that is going in the lab. So herein we have these microneedles where we have loaded these proteins in these microneedles, which are further complex with the nanoplexes and then loaded in the microneedles. So this is all of what we are doing and how we are utilizing the nanotechnology based approach for delivering this gene editing tool. And I would like to acknowledge the collaborators who helped us in designing different components in the research. Dr. Debojyoti from CSIR IGIB lab, IGIB, and Dr. Vivek Singh from LV Prasad Eye Institute, and Dr. Shiva Prakash Ramalingam from CSIR IGIB. So the student who has mainly worked on these projects is Dr. Deepak Sahel, who were in his PhD, and right now he's in doing his postdoctoral studies. And of course, nothing could be done without the funding support. So we have got the funding from the Department of Biotechnology, who had supported us in conducting most of the work that I have shown in today's talk. So I'll be more than happy to collaborate, to discuss various avenues, and just be in touch. And thank you, everyone. And I'm open for any questions.
Thank you, sir, for this informative session of yours. And now the forum is open for the question and answer session. Rather an interactive session with the honorable speaker of this technical session. Anybody offline? Can I request? I would request you rather to please move on to the stage so that sir can also hear you all.
Sir, I was really amazed by your research. So I should congratulate you for that. So, as it is my personal experience, with that, so as you are claiming that corneal dystrophy and retinal dystrophies can be treated with this CRISPR-Cas technology. So, sir, it would be definitely a good experience if you can share any clinical evidences for that. And if it is so, it would be definitely a breakthrough for many of those that are suffering from that.
Yeah, I mean that's a pretty good question that you have asked on the clinical side. So for particularly these retinal dystrophies and clinical and for these corneal dystrophies, there have been studies that are being conducted, but till now none has been tried on directly injecting them. But what we are trying to do is using a system where the CRISPR editing could be done by injecting them either the intravitreal or by applying the topical for the corneal one. So still we are looking forward to taking these systems for the clinical testing, but that's a long way to go for the systems to come to the market. But using other vectors, which are viral based vectors, and doing the editing outside the cells, for example, taking the cells out, then doing the editing, and then implanting them back. So first the cells are taken, iPSCs are created, editing is done, and then they are implanted. So that kind of a strategy is already in where certain clinical studies are already ongoing. But for this kind of a strategy where injections are done, still we are looking forward for this. And maybe in the future we can see some of the therapies do come in the market. But as I've said, it's a long way to go for these systems, and a lot of research is needed.
Thank you, sir, for elaborately answering this question. We don't have any questions till now as of online. So I would. Okay, sir. Right now we could see few queries for you on the chat box. Sir, I am going to address one of them on the behalf of Sivangi Verma. Good afternoon, sir. Are there any promising advancements in crossing biological barriers, such as the blood-brain barrier using nanosystem?
Yes, indeed, there are. In fact, a part of research that we have done with brain cancer, where by modifying the surfaces, we have seen attaching the cyclic RGD and then crossing the blood-brain barrier is already reported. And there are other surface modifications that have been done, say coating your nanosystems with vitamin E TPGS that can also enhance the blood-brain barrier. So reports are there, definitely, and you can just dig out some literature on that. There are already reports. But another strategy that I should bring up is delivering the drugs directly from nose to the brain. That has also been now very well explored. That you may also look into if you are targeting the CNS conditions.
So the next question is, how can we minimize potential toxicity and long-term effects of nanocarriers in biological system?
So minimizing the long-term effects. So first thing is the system that we are choosing. In the research that we have done, herein we are having a completely biodegradable system, and that could be removed, does not accumulate in the body. So whenever you are selecting a particular system, a nano system, you need to consider its biodegradability, and secondly, the biocompatibility. So an ample of studies, protocols are there which we need to look into, that we start with the cytocompatibility studies, the interaction with the blood. So those studies are there that we need to identify and do. But what could be done, that the literature could be used, a system which are already established, they could be looked first to be taken forward, such as polyesters are there, polycarbonates, polyanhydrides. So these polymers are having already few products are there in the market that have shown potential. So if you look on to these products and create these nanosystems, and then definitely there is a high chance to avoid the toxicity issues which are associated. But if you are designing a new system, definitely an array of toxicological assessment has to be done before moving forward.
Thank you, sir, for addressing all these queries so elaborately. And with that, we come to the end of our pre-lunch lunch endeavors for day 2. And I request all our esteemed speakers, dignitaries, and faculties to move to the faculties and all the revered guests to move to the VIP lounge for lunch. However, the lunch for offline delegates have been arranged at the back of this auditorium. But prior to that, I request every one of you to please gather in front of our auditorium for a group photograph. And from there, I request everyone to disperse for the lunch. We are definitely going to resume our offline oral presentation competition right here at the auditorium, sharp at 2 p.m. So those who have opted for offline presentation, I request you to finish your lunch priority so that we can begin our offline presentation on time. Thank you, sir, for joining us online. And thank you for taking time off your busy schedule and with your presence. And we are definitely looking forward for you to join here in the offline mode sometime when we again start for some other such events. Thank you. Thank you for
Sir से बोल दीजिए hard copy we will send.
And we are going to send you the hard copies of your certificates and other elements from our side. Thank you for being here with us. Thank you.
Yeah, thank you so much.
Thank you. I request everyone to please first get assembled in the front of our auditorium for the group photograph. From there, you can disperse for the lunch. I request our online attendees also to resume sharp at 2 p.m. to encounter the different research and review articles to be presented over here by selected participants on Zoom platform. we are going to resume the post-lunch endeavors. The first being the oral offline presentations. I request all the selected participants for oral post offline presentations to please gather here in the auditorium for the final attendance. I request all those selected participants for oral presentations offline to please gather here in the auditorium for their final attendance.
सर के पास बैठ जाते हो सर और मैम के पास
पेन है क्या? पेन
एक दिया है ना इसको बस होगा हाँ
सर वो सब दे दिया बैक पर डाल दो।
Dear delegates, we are going to get started with the oral offline presentation. I request all those participants, those who are interested for oral offline presentation, to please be seated in the first row, left-hand side of auditorium. I am now reading out the name for the final attendance. Deepak Mishra. Deepak Mishra, do we have him?
Maybe take class.
Dr. Priti Mathur. Dr. Priti Mathur was here with us. Others can be tally talk. Dr. Priti Mathur was there with us. Vaishali Kaushal. Do we have Vaishali Kaushal? Vaishali Kaushal. RNA loaded nanocarriers for the management of brain diseases through nasal delivery group. Please be seated in the first row on the left hand side of the auditorium. Samir Beg. Do we have Samir Beg? Do we have Samir Beg? Others? No. Safa Kalam. Karuna Sankar Rathore. Do we have Karuna Sankar Rathore? Others? Do we have Karuna Sankar Rathore? Formulation and evaluation of ketoconazole. Aarti Paras. Do we have Aarti Paras? Aarti Paras. We don't have Aarti Paras. Others? Others, do we have Aarti Paras? Do we have Dheeraj Halwai? Do we have Dheeraj Halwai over here? Subhangi Jarawat. Do we have Subhangi Jarawat? Shivanshi Chauhan. Do we have Shivanshi Chauhan? Please be seated in the first row. Zoha Siddiqui. We have Zoha Siddiqui. Monica Maltra. Archit Akil. Archit Akil. We have one additional participant, sir. I would request you to kindly include the name. Okay. Arpita Singh. Arpita Singh.
Participant are Vaishnavi.
Vaishnavi. Who's Vaishnavi?
Second.
Okay, she is present. Vaishnavi is here with us. Added. Ma'am. Can you speak on
One more participant is asking to join.
You want for oral presentation? the feedback. Here we are begin with our client oral presentation competition. But before that, may I now take this privilege to introduce the honorable judges for this oral presentation competition of line board. We have Dr. Tariq Mahmood, Professor of Department of Pharmacy, Integral University, Lucknow, as one of our esteemed judge. And we also have Dr. Arya Lakshmi Mari Sethi Ma'am, Assistant Professor, School of Pharmaceutical Sciences, Delhi Pharmaceutical Sciences and Research University, New Delhi, as the second judge for our presentation. Before we move ahead with the presentations, may I take this honor to introduce briefly Dr. Arya Lakshmi Ma'am. She was born in Andhra Pradesh, India, and she is currently working as an assistant professor in the Department of Pharmacology and Phytochemistry, School of Pharmaceutical Sciences, Delhi Pharmaceutical Sciences and Research University, New Delhi. She completed her master's in pharmacy in the year 2011 and doctorate in 2021 in the Department of Pharmacology and Phytochemistry, University College of Pharmaceutical Sciences, Andhra University, Visakhapatnam. She is having 13 years of teaching, research and administrative experiences and has guided over 35 B Pharmacy and 15 M Pharmacy students till date. She was awarded Best Ongoing Student for the year 2005 and 2009 and scored state first rank in AURCET 2017. She has published over 33 research papers and over 45 book chapters, 9 patents of India and 2 international patents to her training. Thank you, ma'am, for accepting our invitation and be the judge for this oral offline presentation. I request Dr. Anuradha Mishra, ma'am, Director AIP, to kindly felicitate Marisetti ma'am with a planter as a token of our appreciation and gratitude. May I now have this honor to introduce our honorable guest for being the judge of oral offline presentation, Dr. Tariq Mahmood Ansari, Professor, Faculty of Pharmacy, Integral University, Lucknow. He is having more than 15 years of teaching, research, and pharmaceutical industry experience, and he is presently holding the position as Deputy Director, Career Counseling and Guidance Cell, and Chairman, Institutional Animal Ethics Committee at Integral University. He was also the former head Department of Pharmacy at Integral University. Sir has attended 10 UGC program of academic literacy and has taught numerous publications in international and national journals of review. So we are really glad to have you today with us, and thank you for accepting our invitation and grace this event and being the judge of this offline oral presentation competition. Thank you, sir. I would now request Professor Dr. Anuradha Mishra, ma'am, Director AIP Lucknow Campus, to please felicitate our honorable judge with a planter and request all the dignitaries to join me in this felicitation. Thank you, everyone. Without further delay, I would now read out the criteria for evaluation of oral presentation competition. Dear participants, the duration will be 8 minutes for your presentation, including the interactive session. You will be evaluated on the basis of the content, overall content of your slides, your presentation, and the question and answer session, the way how you defend.
I think this is that you divide that 8 minutes into 5+3.
Okay, sir.
5 minutes would be minutes for presentation and 3 minutes for review. The
Yeah.
question from this as well as from
Yes.
any other base one.
Yes, sir.
Yeah, maybe from this.
Yeah, that's true. It's 8 minutes, including the interactive session. So after 5 minutes, you will be given total 5 minutes to make your presentation. After that, you will be informed and you have to stop your presentation after 5 minutes. And we'll move for your interactive session. So now let's get started with the first presenter, Mr. Deepak Mishra from MIT University, Uttar Pradesh, Lucknow. He is a SRF of ICMR and is presently working on cognitive impairments and exploring the beneficial effects of selected series for this.
Good afternoon, everyone. Here I am Deepak Mishra, SRF ICMR project, working under Associate Professor and Principal Investigator, Dr. Lucy Mahapatra ma'am, on the ICMR funded project, file number as mentioned. So today I am presenting a part of our ICMR research project, which is evaluating the effect of Bacteria hornemoni ethanolic extract on cognitive impairment in aged rats. So about the background, the basis of this research was to tap into the marine resources, which has still been untapped resources. So as it has been visualized that aging is turning into a burden for many countries, but it has also been seen that the countries that has higher life expectancies, such as Japan, Korea, etc., they have higher life expectancy because they have seaweeds in their food routines. So this was the basis of the research. And that's why we have used the seaweed extracts for treating the cognitive impairment in aged rats. This is the basic pattern that evolves during the brain aging. The novelty of our research was that there are still very less number of FDA-approved drugs for cognitive impairment that targets aging population. So the suitable formulation needs to be evaluated for the same. And the objectives of my work would be to study the neuroprotective and nootropic effect of seaweed and its isolated products in vivo aged animal models. Alongside, we also tapped into the mitochondrial biogenesis effect of the same. These are the methods that were procured for the extraction of our seaweed extract. The grouping of test animals were done into 5 of these. The first one was normal control. That included young Vista rats, that is 6 months old, and the other were aged rats that were included as per their treatments. That is, age control were not given any treatment, and the standard group was given with piracetam, which is a FDA-approved drug for cognitive impairment, alongside low dose and high dose of HER, that is hydroethanolic extract of Bacteria hornemoni, 200 mg per kg and 400 mg per kg. Both were given orally. These are the certain parameters that were evaluated during the research. The results are shown in other slides. So the first of all, the percentage yield that we showed was 8.78% for this particular plant extract, which comes under rat seaweeds category. Then the body weight, feed intake, and water intake was properly monitored during the tenure of 4 weeks, which was found evidently significantly improved during the treatment in the treatment groups, as evident with these graphs. Next, the cognitive impairments that included memory loss in the aged rats, and that was found to be improved in the water maze and novel object recognition test. And these were done to evaluate the nootropic effect of the HER, that is hydrolonic ethanoic ester of rat seaweeds. Then after that, the oxidant markers were also evaluated. That is MDA and GSH. We all know MDA are found to be elevated in the aged rats because of its oxidative stress properties. And on the contemporary part, GSH is found to be reduced because it is an antioxidant marker. So which was found to be improving in both the cases. Next, we tried to form a mechanism of action of this work, which was the mitochondrial biogenesis marker was highly improved in the treatment groups, which was evident with our research, with our graph. As we can see, the SIRT1, PGC1 alpha, and cytochrome C are significantly elevated in the test groups, that is HER 200 and HER 400. These are the histopathological evaluations. So H&E and Congo rat was evaluated to check the hippocampus of the aged rats. Okay, thank you so much.
Just conclude you. Just conclude.
So the discussion is
Not decision.
okay. So the future perspective of this work would be definitely to formulate these seaweed extracts. We have 4 seaweed extracts. We can target the main bioactive components and make a proper formulation, which can be converted into a marketed formulation. So it can benefit the higher aging population as well. Thank you so much.
Okay. That project is still going on or it's complete?
It is on the verge of completion, sir.
So, two questions in my mind and just my career. The first one is that what is the rationale behind using the economic extract of that when you go for the cognitive impairment?
So first of all, the blood-brain barrier, the blood-brain barrier is easily accessible using the hydroethanolic extract.
Why you extract the economic extract of that?
So first of all, it is in many of the reviews, it has been found that it is a bit safer than methanolic extract, and it was found to have a more yield in comparative to other extracts using other solvents.
So how do we ensure that all solvent has been removed?
So as we use the rotary evaporator, after that we also utilize the life line.
And that is basically the age model.
Yes.
So what is the criteria of that one? We selected, go to the protocol table. There is the local information. Exactly. So we have taken normal rat as 6 months old.
6 months old.
And age control, we have taken the 18 months.
Yes, sir.
So why 6 month old rat is taken as control? What is the rationale? And why 18-month old rat is taken as OH control? What is the rationale?
Because there is a correlation between rats and us humans, that our 20 days is equivalent to their 1 day or approx. So this builds up our notion that 6 month old rat should be a younger one, that we can say 18 months old in us, in humans.
What
And
is the average of star rat? You taken the star or the
Vista rats.
what is the average age of star?
Average age, sir, about 3 to 4 years. 4 years.
4 years.
Average, we can say, is 4 years, sir.
No, take a thought. Okay. I think. And the second thing is that because that is your disease group or you can say toxic group. You have doesn't put any toxic substance to induce the cognitive impairment. So what is the inclusion criteria of that animals? How to check that the cognitive impairment take place in that group? Take you both for the treatment and others. So what is the total duration of that study?
So 4 weeks.
4 weeks.
In aging, sir, it is already found evident that they have a issue with the cognition.
I understand you taken the normal control, the 6 month old rat, and disease control, the 3 month old rat, and you started the study of 4 weeks. But at the starting stage, how can you assess that in that second group, the cognitive
So
impairment?
histological evidences were seen. We first of yes, sir. We had visualized using the histological evaluation in normal control and age control.
In starting,
We
What is
also to
what-
verify it, sir.
think of that, for starting the starting phase, for histological, you want the tissue. So it means that you simplify the animal in starting, that's what you're saying.
So just to verify it, if whether there is a issue with that or not.
I think you have confused between the protocol,
Yes.
okay? I want to know that second group, in the second group, age group,
Mhm.
there should be the cognitive impairment, then the study is started, then you
Yes,
have to
yes.
second group and different group, so that the cognitive impairment is controlled now, okay?
Yes, sir. For that,
So for that, what is the parameters?
water maze and
Not at the end of the study, in the starting. That's why then you take that animal, na?
Mhm.
Otherwise, normal control animal and the second group animal, they are the same.
yes, sir.
Okay.
So novel object recognition and water maze can be also utilized for the same.
So where is the result of that study?
Yes, sir. Yes, sir. Included. Yes, sir. Yes.
That is the result when your study is completed. I want the comparison of only two groups, the normal control is the disease control, or you can say age control, where you showed that the normal control animal they have the normal one and the age control have the develop the cognitive impairment. Then you further move on that study.
Sir, this is a part of our research, sir. Our research is very vivid, sir. It is just a part of our research. We have done all this, but the data are not present over here.
Okay, but before that is, before that, you mentioned that one, that is the actually, you can say significant level between that, okay? Okay. And you have targeted, I think site one.
Sir,
Okay. And for in antioxidant, you estimated ESH. So, for that cognitive impairment or any such type of memory impairment model, GSS is better or glutathione reductase is better to estimate. What is the difference between total glutathione and glutathione reductase? You are sodium solution or pharmacology?
pharmacology.
Pharmacology. This then is the answer.
Yes, sir.
Okay. You estimated total GSS.
Good.
Okay. And in some paper you find glutathione reductase.
Yes, sir. So, yes, sir. Okay.
Yes.
Glutathione reductase is an enzyme that is utilized for the glutathione antioxidant profile.
So when you pair with MDA, then it is better you estimate glutathione reductase. It is more relevant with MDA.
Yes, sir.
In fact, of total glutathione.
Okay.
There are other sources of glutathione also.
Yes.
Okay. Okay. And
Good work. So how you select CV in your basis on international?
Yes. There were basically there are three types of seaweeds that are red, green, and brown. So we had a lot of 5 seaweeds, out of which we first of all the screening were done. Out of those, 2 were selected in the last year to evaluate the animal parameters. So ma'am, it was collected from Chennai and it was procured from there only. It was authenticated by CDRI. Yes, Lucknow.
What mechanism you develop So you take in the economic extract, right?
Yes, sir.
So economic extract, what are the phytomolecules in present? What are responsible for these activities?
Yes, ma'am. Right. Ma'am, there are several minerals which are already available in the seaweeds, as it is procured from marine resources, and marine resources is abundant in minerals. Sorry, ma'am. Yes, ma'am. We can say sodium, calcium, potassium. These are all found in abundant manner. Not only minerals, ma'am, it also contains polysaccharides, polyphenols. There are certain other different secondary metabolites.
Constituent of that seaweed, which is responsible.
Yes, ma'am. Right. Yes. So the phycoxanthin is for polyphenols, and for polysaccharides, it is yes, ma'am. Phycoxanthin was available. Phycotannin, exact name I cannot remember right now, but it is evident with us. And polysaccharide, it was found to be lignins, that they were also found evidently in this.
How you pre-treat of this seaweed? This is very important, right?
Yes.
So how you treat this?
Sorry, ma'am, not
Treatment of seaweed. Seaweed you have to eat and reach amount of sunlight. How you have to pre-treat seaweed?
I have no idea.
First you collect it, right?
Mhm.
Then you have to go for the extraction. Powder. So how before we have to go for the extraction, we have to do the pre-treatment.
Okay.
What you did? Where you did and here only.
No, so pre-treatment was done here only. So different tests were also performed for that.
What did you
I'm not
observe?
got- I'm not able to get the question. Pre-treatment in what context, ma'am? Mhm.
Soil.
Okay.
Plants you can grow. But how we have to do that?
Yes, sir, ma'am, that was not done over here because as we have directly procured it from someone.
Acute toxicity studies you did, right?
Yes, acute
So
toxicity.
they rats or according to which guidelines you did the dose?
I have included it. 625.
You selected dose.
Yes, ma'am. The acute toxicity study was done according to OECD guideline. It was found to be the 2000 is the rat. We took rat.
You have written, you have not performed the toxicity. Dose determination study is different and active toxicity study is different.
Okay.
So what you perform? You perform only dose determination.
We also performed acute toxicity study.
And again, you perform active toxicity also.
Yes.
Where is the data?
Sir, everything cannot.
So basically, I think what that you perform the dose determination study by OECD for
4 to 5 years.
Next year is not that.
Yes.
From that you determine the dose only.
Yes.
Okay? Because there is, I think, no need of performing toxicity law. That plant is not reported yet previously. It is reported in any other.
Yes, it is reported for several days.
But you don't need that to doses.
Yes, for doses it was performed 4 to 5 days.
And but the question is that what is the rationale of that doses? For that one from that.
Yes, sir.
You find one is the lower dose and one is the higher.
400.
Okay.
1/10 and 1/5 was taken.
I have only one question. Your targeted compound was phenolics, as you said, right? So how you confirm whether phenolics came in your extract or not?
So we performed HPLC for that, and also we performed that pharmacognostic evaluations for that to whether check it was there or not.
Actually, see how we will confirm whether phenolics are there or not. So what markers you have used over there? The SPLC is not the technique which will give you the quality indication without the use of marker. Or if you have conducted pharmacognostic parameters.
Flash chromatography was also done.
It is not applicable. How you will confirm? it is not containing the terpene type of compound as you said, and it won't come in ethanolic extract as well. So whether your samples are rich with phenolics or not, how to confirm? Because we have done the extraction process, but what about the confirmation of
So
the?
as per the literature, IR is used, sir, for phenolic and polysaccharide rich seaweed extracts.
You can't,
Yes, we cannot confirm, but at least we can get the notion that whether it is there.
very simple procedure is whatever samples you are getting after extraction, just go for phytochemical test.
Yes, sir. Efficiency test was also performed. General test, yes, that I was selling a pharmacognostic test that I was talking about. Thank you so much.
May I now request Vaishnavi Garg to make her presentation. Request you to kindly stick to the time.
Good afternoon, everyone. My name is Vaishnavi, and today I'm going to present my research work on volatile-induced neurotoxicity and identification of potential neuroprotective agents. As all of us know that volatile organic compounds are quite toxic, and few sources of volatile organic compounds are oil paints, thinner, grease, etc. Actually, this research is inspired by a real-life observation. We all know renovation is going on in our university campus, and I have noticed that these workers being exposed to VOC for like a huge period of time, for a long-term exposure. And this led me to study about the neurological damage in them because of this VOCs, and I try to identify the primary solution that would be accessible to these affected population. Now, further moving on. As I have discussed, the common VOCs which I have took for my studies are benzene, formaldehyde, toluene, acrolein, and styrene. Now, further moving on. My objectives are to identify the key genes associated with VOC-induced neurotoxicity, to analyze the impact of SNP mutation in hub genes, and to explore the potential of phytochemicals as a neuroprotective agent against the protein expressed by the identified genes. Now moving on to the methodology, I have identified the individual VOC-related genes by using the databases such as GenCard and CDB database. Then I have extracted the common genes by using the Venn diagram. Further, I did the PPI interaction analysis through cytoscape and found the hub genes with the help of cytohubba plugin. Further, I extracted the SNPs with the help of LCBI database. And then I conducted the mutation analysis study with the help of CIP mutation tester and Polyfen2 tool. Further, I did the regulatory and GWAS studies to study the impact of the mutations or neurological conditions. And then I checked the binding affinity of the potential phytochemicals against the protein expressed by these identified genes with the help of molecular doctrine. That was the workflow. Now moving on to my results. Okay, so there were 106 genes that were affected by formaldehyde, benzene, styrene, acrolein, and toluene altogether. I have extracted them with the help of Venn diagram tool. Now this is my PPI network which I have prepared with the help of Cytoscape. I have found the hub genes with the help of cytohubba plugin, and those hub genes were JUN, CXCL8, and TP53. Now further moving on SNP analysis, this is comprehensive self-explanatory results, which is showing that the mutation on JUN at particular position were deleterious. Further, the mutations in CXCL8 at the given position was found to be deleterious by the scores of the tools. But the scores of TP53 were not that good, and it was found to be tolerable. The mutations of TP53. Now moving on, I have found by doing the regulatory studies with the help of GWAS and Regulome that GUN and CXCL8 significantly influence the risk factor in neurodegenerative disorders. Deregulation activates neurodegenerative pathway, notably in Alzheimer's and Alzheimer's diseases. GUN and CXCL8 mediate inflammatory responses linked to neurotoxicity. This is the bulk tissue expression in brain of GUN. For CXCL8, bulk tissue expression in brain. Then I constructed the phytochemical library with the help of PubChem database, and I compile it with the Lipinski's rule of 5 with the help of Admit Lab 2. This is my phytochemical library, which is giving all important informations about the compound, source, smileys, CID, and further I have evaluated with the help of tool that it is being accepted by Lipinski's rule of 5 or not. This is the further extension of the library. Further, I evaluated each phytochemical on the each criteria of Lipinski's rule of 5. Yes, ma'am. This is the docking study. I extracted the structure of the protein with the help of PDB. And I have docked the structure or the phytochemicals against the proteins. This is the most important result that the affinity of protein 1 GUN with tangerine was the lowest, that is minus 8.169, and affinity of protein interleukin that is expressed by gene CXCL8 is lowest with beta testosterone, that is minus 7.9. These are my docking results, which I had did with the help of doctor.
What is the conclusion of your study?
Yes, sir. So actually, tangerine is a natural flavonoid with the antioxidant and neuroprotective properties, and it can be easily incorporated into the diets through the citrus fruits like lemon and chili, making it very affordable and accessible to the exposed population of those workers that are working. Further moving on to the beta testosterone, a plant sterol found in peanuts, again very affordable to the working class, and it acts as a very anti-inflammatory and it counteracts the VOC-induced neuroinflammation. This is the interpretation of my findings.
Okay, Vaishnavi.
Yes.
That is your master's in Vaishnavi?
So I am a bachelor student.
That is your bachelor? Project work?
Yes.
Okay. So basically that is totally the in silico approaches in calculation of the study.
Yes.
And we have used several software,
Yes.
starting with the gain expression finding
Yes.
and lastly to the branding.
Yes.
Okay, so one by one, quickly go through, summarize your work in 3 steps. First, you do what? You do the mutational analysis of the ones.
So firstly, I identified the genes which are affected by those VOCs.
Use.
And then, sir, yes.
From there, you will find out which basic genes.
So there are some databases, verified databases like GenCard and CTD database, and I extracted my genes from those databases.
And then you move to the
So
second part of your
Yes.
study.
yes,
And you perform by this software, working software. By,
so after yes, sir.
okay. So then you find out, go to that table, the binding energy table.
Yes, sir.
that or that one. Yes. So that is, I think, ultimately the conclusion of the, if you want to say that derivative, they have the value energy or kinetic and the beta sequence strong from and I think that is not new one. Everybody know beta there is a very classic teacher of beta sequence already reported which says that there are very good findings for with interleukin and TNF alpha and all that markers which are responsible for the neuronal damage. Okay. So I think the derivative is the only one which you can so that is the finding of it, not that beta is it is already reported for that one. Okay.
Yes.
So what is the limitation of that study?
So the main limitation.
Because I think when we go for that type of cerebral study from a database or a data bank, we find out certain one expected or certain one and select some target to check the binding resistance. So many things have been left out. Okay, many things you can not include everything. There are many other targets which are more important than either 6 or 10. Okay, which you can select for that neuroprotective agent for that same targeting and beta sequence. Then the vice versa, you saying that there are many other compounds you find the best binding compared to that targeting. Okay. So on the theme of that one, what is the limitation of this?
So as you have told that we have to work on like more compounds, and the main limitation is that we
What is the definition of more in silico? Near what is the minimum database you require for any observation?
so there's
To make any observation or to prove any hypothesis? Like in the in vivo study, there is a certain number of universe or whatever observation are required for basic stats. The same fundamentals in the applied in silico study also. So it is you should expose how much number of database.
So you are asking about the genes?
Yes.
Gene database?
Starting from genes,
Okay.
then the target. So
Okay, sir. So basically about the genes, I want to tell that the GenCard NCT database is already a very extended and well accepted database. I have took more than 2,000 of genes for individual, and then I have found the common 106 which are affected by like all the VOCs which I have
that's all good.
in all the like all 5 of the VOCs.
Okay.
What basis you selected these samples?
Ma'am, I actually reviewed the already well cited research papers. I read many research papers which are well cited by different researchers, and I extracted a lot of phytochemical. Although there were like or more phytochemicals, although I can't include all in the P1 PPT, but all basis. On the basis like they are already cited in the well-known research papers, and I look for them in research like different resources, like different databases.
Do you know is it not available for the phytochemicals?
Ma'am, like if I have to make a drug out of tangerine, or like if I have to implement further it in the studies on making drug, then I have to check, I think, the schemes for that.
And I think so we would at the level of bachelor's at the group.
Thank you, sir.
I would now invite Dr. Vicky Mathur ma'am to make her presentation.
Good afternoon, everyone present over here. Myself, Dr. Preeti Nagpur from MIT Institute of Biotechnology, presenting my copyright work that is 7 color of Brevure as a diagnostic tools. Next, please. So, natural resources, we least concern our for the natural resources like light, heat, air, soil, flora, and fauna, and most least about the colors of nature. Next, please. Why I am saying that the colors could be used as the diagnostic tool, because now for the so many instruments are coming up in the market, medical market. So least concerned about the colors. Although 30 years before, just remember 30 years before, 20 years before, when there was no sophisticated instrument, doctor used to see the color of tongue, color of your urine, and the color of your fetus. Stool. Next, please. Next, yes. So, this is my last, for the last years, I am working with my colleagues on the colors. So, in this concept of process, I have also seen there are the sequence of the vibrio. Same sequence of the vibrio is in your diseases also, like violet, epoxy indigo, blue band syndrome, blue color, Renault phenomena. That is the fingers are the blue. Next is green color for the there are many diseases for the green color, but mainly it is the jaundice. High level of jaundice converted the skin to the yellow to green. Next is yellow. Yes, of course, it is for the jaundice. Orange is also characterized by your health for the disease, keratoconema disease, and the hemangiomas for the red color, red patches on the face. So, next is, so why this is the vibrio, color of the light is in our body. And sometimes color is the means changing the color with the change of the disease complexity. Mainly for the bruising information, albinase, color of eye, and jaundice. Colors are changing. Next, please. It is because my study found that it is because we have same vibrio in our body also. Same vibrio. Yes, violet, indigo, blue, green, yellow, and orange, and the red pigments in our body. Retina having violet, blood having indigo, then again the retina having blue, blood derivative and this is the molecule having the green color, yellow color, orange, and the red. These are the there are some molecules within us which shows the same sequence of the vibrio. Next, please. And we have approached the bioinformatics tool, and I was surprised to see that the oxygen producing chlorophyll is the structure of the oxygen carrying molecule, that is the blood. Blood is green, blood is violet, blood is indigo, blood is green, blood is yellow, and blood is orange or the red. Same structure also there, same structure. What is the chlorophyll in the plants? The same structure in the animal's blood also. There are the 7 color of the animal blood. Next, please. For this study, I have also gone for the phylogenetic study, and the maximum likelihood method is used. It is based on the GTP method based model. An enzyme, copro-4-5 gel, is found to be in the leaf, in the chlorophyll molecule, synthesis for the synthesis, and for the oxygen carrying molecule, that is the blood, in all the animals. Next. Same is with the plants also. The plants.
Book for conclusion directly.
Okay. So next is yes. No, before that. So color is actually we see our biosphere horizontally, not the vertically. Vertically is evolution, and we less focus about the evolution. Next is color is flowing. Color is flowing in our universe. It is very strong. Next, please. Next. Here these are some copyright. And the color got be, we know color only for the creation and the painting of walls or the paintings, but it is the energy, it is the indications, it is the genetic flow, it is the cyclic, it is the healing property, medicine. Color is the macro-biological effect, color smooths the nature.
Okay.
So, for the, means it can be help in the pathology. It could be the diagnostic tool. It must be mentioned in the diagnosis. Thank you.
Okay, thank you. I think very interesting topic for family discipline. I'm going but I want to know from your side, in that whole study, which part will go for patent idea? I think that last part.
It's all copyright. It's all copyright.
That starting part, that that's information that color is for that disease, that is the general
No,
knowledge.
What is for the list of before
That is not, I think, your study mentioned, that is the starting slides, the color and the disease. That is the actually, that is the general information of the pathophysiology, I think.
that is
So
okay.
what I want to know from your side, that whole presentation, which part to go for IPR?
For IPR.
Yes,
That is
copyright.
not, yes, yes, fine. For this presentation only, although I have 13 copyright for the colors. Actually, this color is need to focus much.
And yellow is for jaundice and green for albinism and red for angioedema. That is report. That is remove for
I agree with you. I agree with you.
That's what I want to say that go to the last slide where we have use the bioinformatics tools for coding
For the
item.
coordination,
Yes,
it is
I
so-
think that part is the part which I will have to use it for IPR basically.
Patent, next slide here, before that, there is 13 copyright, not the patent. Patent could not be done because it is not a product, it is study on.
Yeah, copyright.
It's
Okay.
copyright.
So, we have, your conclusion is also the same, and you want to say that you as diagnostic tools.
Yes.
Include
It
that
should be,
your
it should be. Very
include.
properly,
No,
I
and
suggest.
what's the basic limitation of that study? If I ask you that question, if we recommend that review is a diagnostic tool and should be read to all the pathology students. What is the problem in that one? What is the limitation of that one?
Limitations.
Yes.
Because it is
The
not-
first is that there is no, that clearly represented that, data, of the color mapping with the certain disease. But it varies actually geographically also. That is not applied the same with that Asian population or the
Yes,
African
I agree.
population
It
or
is
the.
environment. There is
Yeah,
no environment. Yes.
Yeah, there is a first limitation.
Yes.
And the second limitation is that the big viewer from that violet to the red, all that.
This is the copyright, yes.
That's okay. Okay, from that video to red, all that color, that is not attributed to that disease which you listed that one. There are so many others for it.
Yes, this is lot of diseases, sir.
Very much.
I would give only few.
That is the actually one
Yes,
limitation
I
or.
will give to ICMR also.
Yes,
So please include the color in your syllabus.
that's what.
Because there is no content in the medical science, this is the color, importance of the color, also the body having. Thank you.
Oh,
Any other question?
Thank you, ma'am. May I now request Vaishali Kaushal to make her presentation?
Good afternoon, everyone. My name is Vaishali Kaushal. Today I am going to present my oral presentation on topic RNA-loaded nanocarrier for the management of brain disease through the nasal route of the delivery. So the RNA-loaded nanocarrier are ability to encapsulate. Sir, I have included review work. RNA-loaded nanocarrier are ability to encapsulate and protect the RNA from the degradation, and they can enhance the transport to across the blood-brain barrier. And these carriers, they have to enhance the stability and viability of the RNA therapeutics, and they can ensuring to precise the delivery of the affected tissue while the minimizing the systematic exposure. The nasal mucosa and its vascularization and olfactory route are the direct pathway for the entering the drug through nose to brain. And intranasal delivery of the RNA-loaded nanocarrier are the promising approaches for the managing the brain disease. There are various type of RNAs which are used for the therapy of the brain disease like mRNA, siRNA, microRNA, and shRNA. Rationale, why choose the nanocarrier? Because they can protect the RNA from the enzymatic degradation, facilitate to control and targeted release, improve the cellular uptake and blood-brain barrier penetrations. When the RNA with the nanocarrier, they can deliver through nose to brain, they potentially benefit to enhance the drug stability, improve their viability and absorption, and targeted delivery, and reduce their systematic side effects. And overall, they can enhance the brain targeting and stability and treating the brain disease. Is the conclusion, the RNA-loaded nanocarrier for the nasal drug delivery, which can treat in the brain disease for the developing personalized medicine approaches and combination therapies and long-term treatments.
Okay. You are master's student?
Yes, sir.
That is your review article?
No, sir.
You are working on that same topic on your thesis?
No, sir.
So why you choose that for presentation? Give me, five, not like three example of RNA-loaded nanocarriers which are used for, and you have mentioned very got a brain disease. Okay.
Yes,
So
sir.
make a table, put here three RNA nanocarriers and any type of nanocarriers for the particular type of brain disease. Name three. That type of neurosome for that disease, for Alzheimer's suppose that type of carrier for that one. Give example of three.
Sir, miRNA
For each disease.
for
That is your topic, na? Actually, in that slide, there is a slide for that one. There is a table for these are the nanocarriers which are already developed.
yes, sir.
Okay, for this brain disease, and especially with the nasal route.
Yes, sir.
Brain target. Okay, and there are certain which are available as the oral one. There are certain which are given by the injectables. Okay, when you go to the literature, you find out that is a very huge topic, it's a vast topic. You can have loaded nanocarriers. Okay, and there are, you mentioned that in the slide there is a n number of types of nanocarriers available. From that, you can practice three examples for particular three brain diseases.
Yes, sir. The solid lipid nanoparticle loaded siRNA for the Alzheimer disease. Or sir, miRNA 132 for the cerebral ischemia.
Okay, sir.
Okay, thank you, sir.
Next presenter, Safat Alam.
Good afternoon, everyone. My name is Shafat Alam. I'm Pharm second year student from Integral University. My presentation topic is AI integrated and nano carrier mediated intranasal drug delivery system for the management of brain diseases. For introduction, I have taken intranasal route because it can bypass blood-brain barrier, and from the route of trigeminal and olfactory route, it can reach to the brain. And nano carrier helps in that because it has a nano size, ranges from 1 to 1000, and for brain, it should be between 1 to 200 nanometer. AI helps in simulation of the human intelligence in machines that are programmed to think, adapt, and learn. Objective to perform literature search related to brain diseases, nasal route of drug delivery, and AI integration in nasal drug delivery, and to report published case studies related to AI integration in conjunction with nanocarrier mediated intranasal drug delivery for the management of brain diseases. Rational. So a conventional drug formulation can have low bioavailability, low transmembrane permeability, and less aqueous and lipid solubility. And if we take oral route, it will have first pass metabolism, gastric irritation, slow onset of action, gastric degradation of the drug, and inactivation by gastric enzymes. So if we incorporate the drug into nanocarriers, it will have enhanced drug solubility, improved bioavailability, sustained release characteristics, enhanced drug loading capacity. And if we give it to nasal route, it will have rapid absorption, it will avoid first pass metabolism, good patient compliance, reduce GI side effects. And if we So, for conclusion, it will have a great potential taking AI, nanotechnology, and medicine together to innovate a new treatment and improve patient care. Call to action, researchers, clinicians, and industry professionals are encouraged to actively engage in this rapidly evolving field to maximize its benefit for patient. It will have a continuous research work and improve AI integrated nasal drug delivery of nanoparticles for brain diseases. Enhanced AI integrated nasal delivery or nanoparticles has shown potential for improving drug delivery to brain in the treatment of brain-related disorders.
Your topic is actually AI integrated and nano carrier integrated. What is the meaning of and here? Understand my question? Topic is AI integrated and nano carrier integrated.
So we have integrated AI and through using nano carrier.
If you remove and from here, then AI integrated nano carrier. What is the problem? What is the use of and here? The first thing. AI integrated nano carrier mediated. Okay. So is there any literature you find out any paper in which some machine learning tool is used to develop certain nano carrier mediated transmissibility system?
Yes, sir. I have found 7 articles, case studies, and I have included 2.
Okay. Have you studied that paper? You find that is case study, not that is a paper.
Yes, sir. Yes, sir.
That is a review paper.
Yes, sir.
Okay. Paper. Have you studied that paper? What is the finding of that paper?
Sir, it helps in optimizing the formulation. Like they have taken 3 input layer and 3 output layer. In input layer, they have taken glycerol monoylate and poloxamol and 280. And output layer, they have taken particle size, entrapment efficiency, and drug release. So using ANN model, they have make an optimized formulation. As you can see that F6 batch have shown a good drug release, entrapment efficiency, and particle size.
Have you gone through the literature of ANM or application method? That is a very broad spectrum. Okay. So it cannot directly said that the ANN model reflects that neuronal network reflect that or mimic that certain problem of the brain or certain disease. Okay.
Okay, sir.
And the second thing is that go very broad in the test material form like nanocarrier related. You will be specific with the disease, not brain disease. Okay. Look for the one disease, target one disease.
Okay, sir.
And then select one nanocarrier,
Okay, sir.
one formulation. Okay. Then go to deep to find out what is the machine learning tool available or AI integrated or AI assisted technique available to develop that formula. That is the way we are going to design your master plan or PhD one. Okay. Otherwise, it is only a PDT, time material or utensils and nutrition material. Okay.
Okay, sir.
Okay.
Organize it, it's not that work.
ANN model, I know.
AI.
AI models.
Software tools, authorized software AI tools. except
AI
the
tools.
ChatGPT.
Okay, okay. ChatGPT,
Except the ChatGPT.
DeepSeek, SciSpace, DeepSeek, SciSpace.
Okay. That's the authorized AI. Next participant is Shivanshi Chauhan. Shivanshi.
Good afternoon, everyone. I am Shivanshi Chauhan, student of M.Pharm Pharmaceutics 4th semester. And today I'm going to present a review work on harnessing Moringa oleifera's potential for wound healing, which is guided by Dr. Richa Srivastava ma'am. Starting with the introduction.
So far methodology, result and conclusion.
Sir, actually it's a review work, so
No issue, no issue. Move ahead. What is that time?
Sir, the studies of Moringa improve wound contraction. So the various studies which I have seen here is the acceleration of wound closure when this Moringa oleifera leaf extract was used, and the antioxidant enzyme activity increased antioxidant levels, the scar formation and promotion of fibroblast, and minimizing the scar formation. So these are like few papers which I have seen.
Next.
The application in medicine and industry, food and nutraceuticals. It is rich source of nutrients like in vitamin A, C, E, and essential amino acids, making it a superfood. Functional foods and nutraceutical benefits also, like supplements to hypertension, diabetes, inflammation, because of it's having rich in bioactive compounds. It is also good as in the cosmetic and skincare industry, good for skin, enhancing the overall health of skin, hair.
So what is the conclusion, Shivanshi?
Sir, if I come to the conclusion.
Yes.
Just. So the conclusion part is Moringa oleifera is a natural herb, which is the antioxidant powerhouse, which is having accelerated wound closure properties due to rich in antimicrobial properties, antibacterial, anti-inflammatory properties, and it is rich in flavonoids. So it is it is having a good potential for wound healing, for accelerating the wound healing. And moreover, apart from wound, it is also seen as a good tree which is having zero wastage, as each part of its tree is rich in bioactive compounds. So that just a minute, ma'am. There is one slide which I've included. Ma'am, you're asking about one particular part. Which part is more effective than all the parts? Ma'am, I cannot quote one part according to the knowledge which I have, but the leaves, its roots, stem, bark, they are rich in bioactive compounds. Different among all psilocybin flavonoids are teaching only among all the flavonoids in which part is more? Ma'am, I don't know exactly, but maybe it's leaf. Ma'am, leaves are containing more flavonoid content, and moringa coffee, chocolate bars, these are all available nowadays in the market. True, ma'am, true. It is available. But the purpose of this review was to harm. Which one is more effective? So accordingly, friends will leave that mark. Ma'am, I can quote one example as I know. Like one of the interviews which I've seen our Honorable Prime Minister Narendra Modi, he was quoting that he makes use of moringa leaves in parathas to make it. That is having antioxidant effect. Not a researcher, right? It's not a researcher, so you have to give the evidence for that. Ma'am,
Okay. And one thing that is like a master's student?
Yes.
First year? Third semester.
Fourth.
Okay. What is the minimum review article required to say that I published that review article and so many number of review article have been studied? Yes, 5, 6, 10, 100, 1,000.
Sir,
how many review articles have you studied for Moringa?
So this 150 plus articles.
150 plus.
Yes. Sir, this article is already communicated in Bentham, and one copyright is also. So few part I have not included because of the publishing thing. That's yes. Thank you, sir. Thank you, ma'am. Zola Siddiqui, the next participant.
Good afternoon to all. I, Zola Siddiqui, am glad to have this opportunity to present at this impeccable conference that is organized by Amity University. And my topic for today is exploring the anti-cancer potential of coumarin derivatives. First of all, next slide. Next slide. So actually, this is from a review article, so I've not included the methodology. And so, can I continue with whatever read? Okay, so first of all, I'd like to have your attention towards the danger of cancer as a disease in our society, because cancer is a heinous disease and it has taken several lives in the past few years and is currently tormenting a lot of them recently. And as pharmacists, we have this responsibility towards our society to develop some anti-cancer drugs that are going to be really effective towards this disease. One such compound that have gained a lot of attention in the recent years is coumarin. Coumarin is basically a benzene and pyrrolidine complex that comes from a flavonoid family. of plant secondary metabolites. These flavonoids are in nature helping the plants to protect them from biotic and abiotic stress. Coumarin has been a promising compound because of its biodiversity and versatility. Hybridizing coumarin with different anti-cancer pharmacophore has given us hope in order to develop some anti-cancer drugs that are going to have lesser side effect and unquantifiable tumor anti-tumor properties. Can I have the next slide, please? Okay, let's look at one such hybridization of coumarin. Here we have taken coumarin and 1,2,3 triazole hybrids. So here we have taken 3 cancer cell lines, one for prostate, the other one for gastric, and the last one for liver. Here we have taken colchicine as the reference drug because colchicine is an alkaloid and is very well known to use to treat gout. It has recently been showing some anti-cancer potential as well in order to inhibit the metastasis of the cancer. The major problem with the cancer disease is its metastatic state. When it achieves it, it is not able to treat it easily. Here we can see that I have derived a SAR from that study. This SAR shows that we can have a hybridization of coumarin, and at the carbon 4 position, it is going to show a selective enhanced property, and at carbon 7 position, it is going to give us a selectively potential compound. Here I have attached 3 compounds from that study. Here, 1C compound has been showing the best anti-cancer activity against all the 3 cancer cell lines. In the very next slide, I have tested a table. Can I have the next slide, please? Here you can see that this 1C compound has better efficacy towards treating cancer in comparison to colchicine that has been taken as a reference drug. So here we have analyzed it through the MTT assay. MTT assay is basically a colorimetric method of cell analysis. I would like to explain how MTT assay is going to work. First of all, we are going to take the MTT substrate, which is yellow in color. At first, when it is involved in the culture of the cell, the living cell are going to convert it into a parmesan crystal that are basically purple in color. So the amount of parmesan crystal present in the culture at the end is going to be proportional to the number of the living cells in the culture. So it is going to represent the site. Okay, sir. Can I have the last? Sir, it is, can I explain this one as well? Can I have the previous one? Previous one. Here I have attested another the hybrid of coumarin. Here we have taken taccharin as the hybridized molecule. The coumarin and the taccharin, both moiety are going to have a number of certain carbons as linkers. Here we can, I have extracted a SAR as well. 6-methylene spacer in the coumarin and taccharin moiety is going to show us optimum efficacy towards the anti-cancer, the cancer. And can I have the next slide, please? Here, basically, I have not tested the whole result because that was a lot to adjust in this particular presentation.
In starting, you said that is a review article, but there are some studies also.
Yes, sir.
So that's not a review presentation. You have included the MTTS.
Yes, sir.
You included that in.
So basically, it's not my work. It is somebody else's work that I have compiled in this presentation.
This is not your work. Give me also 2 questions. Apart from covalent derivatives, give 3 examples which are very popular moiety to explore for anti-cancer. Basic chemistry question.
Yes, sir. So one is imidazole. The other one is the benzimidazole is also very popular.
Very good for anti-cancer.
Sorry, sir, it is very popular for antimicrobial work. Sorry. So I cannot recall this right now because I'm currently working.
And the second question is there, the other arm of that study,
Yes.
anti-cancer potential. Give if there are for covalent derivative, you do not go for anti-cancer, which disease you can target for covalent derivative.
Sir, for combinant. Yes, it's my
And the next question, why CHE anti-cancer still they say have been performed in silico? What is the relation of that one with anti-cancer potential?
Sir, acetylcholine has been given a certain relation with the anti-cancer properties. So,
Where
sir,
and
in the
how?
lung cancer, and, sir, in the lung cancer via the nicotinic receptor.
I want to ask that a very basic molecular pharmacology. What is the relation of ACH with cancer?
The acetylcholine with cancer.
If not, why you explore ACH? Can you yes, what is that?
So 1 minute. Sir, I have to,
Study that.
sir, I have this in mind. Give me just 1 minute to frame it once. So what I've studied is that ACH overexpression has been linked to cancer proliferation and its migration in the lung cancer. So that's what I have studied.
Okay, so.
Yes, sir.
Thank you.
Thanks, sir.
May we now have Monica Moitra, the next presenter. Good afternoon, one and all present here. I am Monica Moitra. PhD scholar at Amity Institute of Biotechnology, and my work is on identification of potential therapeutic targets in gallbladder cancer through network enrichment analysis. So, first of all, I would like to explain about gallbladder cancer, which is the fifth most common gastrointestinal cancer all around the world. Yes. So here I have derived the proteins which are expressed in cancer, and I have performed the protein-protein interaction using the string database. Along with that, I have done the epitope prediction using the IEDV database and docking using CBDoc2 web server. These are the proteins which have been identified in several types of cancers. Out of this, I have selected WEN7A protein. So here we can see the different types of proteins which are expressed in gastric cancer, breast cancer, as well as in gallbladder cancer. These are the ligands which I have identified for WEN7A protein from literature survey. Out of this, using the CBDoc tool, I have docked these ligands with the WEN7A target protein. And here I have got periclosin with the least free energy, and it is the best ligand that has shown the docking presence. Using the Discovery Studio software, I have seen the interaction between periclosin and WEN7A protein. Other than this, I have done the epitope prediction for the WEN7A protein, where the linear epitopes, B-cell linear epitopes, as well as the B-cell conformational or discontinuous epitopes were predicted using IETB web server. The conclusion is that WEN7A protein can be
it has shown the best blocking results with periplosin drug, and hence the Ven7a protein can be assumed as a good biomarker as well as a therapeutic target involved in gallbladder cancer. I'm very thankful to Amit Street of Sir, it is a part of my PhD work.
So, you categorize the identification of potential targets.
Yes, sir.
So from that list of targets, why you have selected only one WNTC? I think, move to that slide. The starting, yes.
Ven7a
Here is also mentioned, why only WNT7? Why not FGFN? If you go for literature, you will find out many different FGFN19. The target for not only for gallbladder cancer, but for the prostate cancer also, which
Yes,
is
sir.
which is very difficult for to treat, and if you can say hot cake nowadays. There are certain cancers which are hot cake nowadays, the breast cancer and the prostate cancer. So, and for that one, FGF19 is very explored. Why have you not selected FGF19?
So till now I have done my research on Ven7a protein.
Okay, that-
Further,
But my suggestion is that on that FNF19 also. Okay. And the second thing is that to go to the binding score, what you use the bioidentical studio,
Discovery Studio, sir.
discovery studio. You have the your version available in your lab, or you have done from somewhere else.
No, sir. I have installed it on my system and then
That is free version of discovery.
yes,
Okay, that is the free version of discovery.
yes.
But my, because you are the PhD scholar, go any lab where the discovery version is available, the latest version, and then you find out that why are you saying that one? You go to the figures on that score. That's same thing. That one. So from that one, I catch that one. That is the free version of discovery.
Yes, sir.
For PhD, but that is not acceptable in your PhD thesis. Okay, so you go talk about it. And explore the bio method for that one. Make a study plan to target at least three. Select three proteins, and from that one, select which one is better from that one. And from that, these are 9. We have selected best 3, and from best 3 you have one, then you conclude in the final PhD one, this target is best therapeutic potential for gallbladder cancer. Okay? Okay.
Thank you. Ma'am, actually, I have selected it from literature. These ligands were are supposed to
Selected chemical or phytochemical
phytochemicals.
already important.
Yes, ma'am. The compound. Ma'am, I have selected them from literature survey.
On which basis?
Ma'am, on the basis of which that they can act as good inhibitors of Ven7a protein.
You are targeting something.
Yes, ma'am. Till now, I have worked on Ven7a protein.
How the standard you are using?
Standard, ma'am, in the sense.
Okay.
Thank you.
Archit Akhil, our next presenter. Archit Akhil.
Good afternoon, everyone. Good afternoon, judges. My name is Archit Akhil, B Pharma first year from Era University. And my topic is oncolytic viruses, oncolytic virus therapy, and revolutionizing cancer care. So it's not changing actually. So cancer.
Next slide, next slide.
Current treatment strategies that are available for cancer: hormonal therapy, surgery, bone marrow transplant, chemotherapy, and radiotherapy, which come with side effects related to them, like hormonal imbalance, increased chances of infection, graft versus host disease incompatibility, impaired immunity, and redness, blister, or digestive issues. Oncolytic virus therapy. What the oncolytic virus therapies? Oncolytic viruses are pathogenic viruses which are engineered or re-engineered to be used for cancer treatment that comes under immunotherapy. And the need for oncolytic virus therapy is that they are tumor specific and they don't actually infect normal cells because of different antigens that are present on the cancer cells and absent on the normal cells. So they can only replicate and infect the tumor cells. The advantages of OB therapy over conventional therapies are low probability of causing generational resistance, which is possible in the use of chemicals like chemotherapy or hormonal therapies. The dose increases with time, non-pathogenic, that replicate only in tumor selective fashion and induce immune response against tumor cells. These are a few recent developments. Imlifidase injection for melanoma. It is the first FDA-approved OV drug developed by Amgen Pharmaceuticals, Amgen company. Neuronova 1 for glioblastoma under phase 1B/2 trials by Calorie Biotherapeutics. Then become to the last Olvi-Vec designed for aggressive solid tumors under phase 3 by Genelex. And here you can see the manufactured and market.
What is the conclusion of your review?
The conclusion for my review is if we engineer and try to use the present pathological viruses for immunotherapy, we can with time we can with time overcome serious side effects and challenges in the cancer therapies that are related to hormonal therapies or chemotherapies.
Okay, Arjun, I think very interesting topic, oncolytic virus therapy, vaccine cancer care, and you make a table here. Go to that table. Where is the latest status of the drugs?
Sure.
That.
This one.
Make one column here where you put that for what cancer it is. Okay?
Okay, sir.
Okay. If that is a drug, that is a component which is here, that is the trial phase. Before that trial phase, there will be a column that suppose that the second one, neuronoma one, that neuronoma one for which cancer?
Glioblastoma, that are separate.
Yes,
Sure.
can mention here. So by that one, it is a very good informative table. And my question is for you that apart from that oncolytic virus therapy, there are, I think, few more very new or novel therapeutic approaches which are under trial for cancer. You know anyone?
Like the one which I have read.
One is viral based.
Vial based.
That should, you should also know that one.
Okay.
Which one?
Vial based, actually, I don't yet know because
Okay.
I'm
Other
yet
than
studying. Drug repurposing is under.
Okay. Okay, I think Archit is in B.Pharm first year.
Yes, sir.
So I think he is the most junior among all presenters because the presenters from the teacher faculty members, PhD scholars and master. So I think Archit meets us all of us.
Thank you, sir. Sure. Thank you.
The next participant, Arpita Singh.
Everyone, I'm Arpita Singh. My topic is Revolutionizing Healthcare, Future Aspect of Neosomes.
Next slide.
The vesicular drug delivery system. Vesicular system are novel means of delivering drug in controlled manner to enhance viability and get therapeutic effect over a longer period of time. Vesicular system are attractive nanocarriers that because their viability and their encapsulation hydrophilic and hydrophobic moiety. There some nanovesicles are present niosomes, liposomes, transferosomes, phytosomes, equasomes, verosome, and cubosome.
Next slide, next.
Classification of niosome. There are three classifications of niosome: multi-laminar vesicles, large laminar vesicles, and small laminar vesicles. Compositions of niosome. There are three compositions: non-ionic surfactant, cholesterol. Example of non-ionic substance span 60, 40, 20, 85, and 80. Twin 20, 20, 40, and 60, 80. Cholesterol maintain the rigidity and proper shape of niosome during preparation. Additives.
Next slide.
Merits and demerits of niosomes.
Go to topic health. Your topic is future aspects of neosome.
Yes.
Revolutionizing healthcare. Go to that slide. Not that in the kind of preparation. Every 6% on that. How neosome.
So this is the main future aspect. Neosome is used in very cancer therapy like ovarian cancer, lungs cancer, breast cancer, prostate cancer, pancreas cancer, colon cancer, skin cancer, liver cancer, ovaries cancer. And various applications: genes factor, nasal delivery, vaccine, and pulmonary delivery, ocular delivery, and proteins and peptides.
Next. What is this?
So then lie laminar vesicle, the LUV formulate where small injection methods in lyopene, drug and loose therapy. The for applicable for blood glucose level reduction and anti-cancerous activity. And small laminar vesicle formulate gingiva through anti-source activity, antioxidant, and anti-inflammatory.
Okay. The advantage of neosomes over other delivery systems when you talk that it is the future of the delivery systems.
This neosome are high value neosome and shows high valuability and
There are many others that have the same biodiversity and neosome. So what are the disadvantages of neosome? Not advantage. What are the disadvantages of neosome?
yes, sir, some problems are instability.
Then why it can revolutionize the healthcare? How it revolutionize the healthcare?
Sorry.
You say that it is the future of the healthcare neosome. Why you say that statement? What is that? What is the cutting advantage of neosome over others?
Sir,
You know any drug which are in market which are neosome from?
the curcumin. The curcumin.
Curcumin.
Yes.
Okay. Okay.
Our last participant, Jay.
Good afternoon, everyone. I am Jay Chaurasia from ITM University, Gwalior, presenting my topic, Organ on Chip. This organ on chip mainly helps in the drug discovery. So let's know about drug discovery. It is a process of identifying, developing, and developing new medication by researching disease mechanism, finding drug targets, and for safety and effectiveness. Basically, there are two traditional methods, animal model and cell culture, which comes through their own challenges.
Then we move to organ on chip now. Then we move to that slide where your topic is started. Yes.
This organ on chip is a small flexible chip which separates by a porous membrane which enables the cell communication, and the extracellular matrix provides the support to the tissue and helps in mimicking the in vivo condition. This organ on chip technology recreates the functional unit of organ and provides the real-time insights of human biology. A key feature of organ on chip is its ability to control the mechanical forces and the condition from outside as per our need. It basically contains 6 parts. The top channel, which provides the airflow, and then second is the epithelial cells, which provides the protection barrier. Third, the vacuum channel, which provides the mechanical forces like breathing and the movement. Fourth is the porous membrane, which provides the cell interaction. Fifth is the endothelial cells, which provides the movement of blood vessels, and the bottom cells, which involves the blood vessels and also helps in the removing the waste material. Next, the experiments which are being done with this organ on chip. First, the recent experiment, the COVID-19. Here, the researchers used this to test the SARS-CoV-2 virus using the lung on chip, where they test how the mechanism of this virus, how it affecting our lungs. Next, the kidney toxicity. Here, cisplatin, which causing the nephrotoxicity, is being tested with this kidney on chip. Next, heart toxicity screening. Here, the doxorubicin is being tested using the heart on chip.
Yeah, go to the conclusion. This slide was the experiment program.
Sir, future uses of this. Sir, the future.
What is the conclusion?
So in last, the conclusion is that it is an innovative technique which can enhance our drug testing. The time we are taking to develop the drug and the animal models and the cell culture models which we are using, which may have which have lots of uh, which have lots of errors, and just to get the efficacy, the proper drug, the efficacy of the drug, we can use this organ on chip, and we can develop the drug very fast and very effective, and we can make our personal medicines as per the need of the patient. Thank you, sir.
Yeah, I think very interesting topic now that organ nuclear path has been discussed nowadays on a very huge scale. You have mentioned example of that also. Is there any regulatory obligation in organic model?
Yes, sir. Yes, sir.
Which committee you need to approval for that one? For which committee you go on for that organic model? Like for animal, you go on for animal. Same thing with that one.
Sir, that is only the limitation. Still, we didn't have the regulatories which to manage this organ on chip, and we didn't have the standardizations yet, but it is in developing phase. So we didn't have that specific, but the CDSCRO and various other organizations are there, which will further.
There is a model which are already applied for the parking. You know that? One such that, okay?
Yes.
And you mentioned the example of 5 diseases.
Yes.
So that is on that one. Okay? Okay. The application has already tried to distribute for that. Okay, that's a good one. Thank you.
Thank you, sir.
Thank you, sir. And with that, we come to the end of the oral presentation competition. Thank you, the participants, and of course, our selected judges. Without you, it would have been next to impossible to conduct this event, sir and ma'am. May I now take this privilege to request Professor Dr. Anuradha Mishra, ma'am, to felicitate our judges with a memento. I first request ma'am to felicitate Dr. Tariq Mahmood, sir, with a memento. May I request you to kindly join us, sir? So, thank you, sir, for accepting our invitation and gracing this event with your presence and providing the connection of oral presentation online. I request ma'am again to felicitate our second judge, Dr. Arya Lakshmi Marisiki, ma'am, with a memento as a token of our gratitude. Thank you, ma'am, for gracing this event with your presence.
All participants were presented the review presentation. Whenever I said that go to the methodology and results, all have the same answer, sir. It is a review article and it doesn't have methodology. So please, it is not that so. Okay, the review have the methodology. Go and study what are the methodology to review the article. Okay, there is a one arm, there is a double arm, there is a helix arm, and the latest one is the meta analysis. That are the processes or types of the methodologies you want to review any topic. Okay, go and study that one. Okay, thank you.
Thank you, sir, for your advice. Respected dignitaries and delegates, both at online and offline mode, it is now time to say goodbye. And here we are in the valedictory ceremony of the Day 2 Extended National Conference, RBPS 2025. I'm honored that we have with us Dr. Neeraj Subramanian Sir, Member Central Council, Pharmacy Council of India, Pro-Vice Chancellor, Sage University, Bhopal, MP, as the Honorable Chief Guest of this event. He is joining us on the online platform on Zoom. May I request our volunteers to make the view on the same? May I request Abhimanyu sir to kindly turn his camera? Sir, if I'm audible to you, can kindly put on your camera?
Yes, you are audible. Yeah, yes, you are audible, ma'am. Perfectly audible. Thank you so much for joining.
Thank you, sir.
Thank you.
So sir is there with us online, and to grace this event, we also have our own beloved and respected Professor Dr. Anuradha Mishra ma'am, Director AIP Health. And we also have other dignitaries and HODs and employees from several other departments of the university campus. We have here with us the honorable judges for oral presentation competition and the honorable speaker, sir, Professor Dr. Ajay Sharma, the first technical session with us for this valedictory ceremony. Thank you all for being here with us. May I now take this privilege to introduce officially to this auspicious gathering the Honorable Chief Guest of Dr. Neeraj Subramanian Sir. He is an esteemed academician and researcher with extensive contributions to pharmaceutical education and research. He completed his M.Pharm and PhD from Department of Pharmaceutical Sciences, Dr. H.S. Gaur University, Sagar, MP, India. Currently, he serves as a professor and pro-vice chancellor at Sage University, Bhopal, and is an active member of the Central Council, Pharmacy Council of India, New Delhi. Dr. Ubhamanyu has played key roles in policy making and accreditations, serving in committees such as the Education Regulation Committee, Professional Pharmacy and Public Relations Committee, and Law Committee at PCR. He has contributed significantly to the syllabus development for UG, PG, and diploma programs, and has served as an expert committee member in AICTE, UGC, NAAC, and NBA. Internationally, he collaborates with the International Pharmaceutical Federation, Netherlands, as a part of the education planning group for the FIP UNESCO Global Pharmacy Education Project. With over 124 publications with a cumulative impact factor over 135, Dr. Ubhamanyu has authored 5 national and 3 international books and 8 book chapters and holds 10 Indian patents, 1 granted, 8 published, and 1 filed, and 1 international patent to his credit. He has successfully guided 7 PhD and more than 25 M.Pharm students. He is a member of various professional organizations, including APTI, ACPI, IPF Netherlands, IPA, CSI, IHPA, CPA, and LASA. He received his several awards, including Principal of the Year 2021, Dynamic Pharma Leader 2020, and Nirali's Excellence in Pharmacy Profession Award 2019. May I now request the Honorable Chief Guest of this event to bestow his words of blessing on all of us. Sir, please.
Thank you, madam. First of all, My heartiest congratulations to Director Anuradha Mishra Madam and the organizing secretary for this wonderful conference, for 2 days conference. And my apology that I couldn't join this conference physically. My sincere thanks to one of my very good friend and colleague, Dr. Abhishek Tiwari sir, who has invited me for this conference and to be a part of this validity program. I congratulate the management of Amity University Lucknow for organizing this conference and the wonderful theme that is recent breakthrough in pharmaceutical sciences. Well, I really very happy to see this conference and the theme which has been selected for this conference, because it is the demand that we have to think, because now we are going from wet lab to dry lab on in all the streams, not on all the streams. So it is very important to to think that how we have to move forward in these directions. I hope during these 2 days conference, a lot of sessions have been conducted. A lot of speakers were there who were talking about innovations in pharmaceutical sciences, the development of drug discovery. But dear friends, it is very important to understand that we have to make a balance between the wet lab and dry lab. Because if we are talking about that, the work which we are carrying out in laboratory and the way we are developing our things onto a pilot level scale up, we have to understand the basics and we have to understand the science. Without the basic research, I think it will be very difficult to understand for the students who are carrying their research at postgraduate level, at doctoral level. We know that right now we have a great facility. In our time it was not available. We have to go for NCL and other laboratories, DRDO, these libraries also to search out the contents. But it is readily available. Now the important part is that how the contents which are available you convert into the basic research and the advanced research, because you have a great literature review, you have a great literature in front of you with the help of the digitization. But now you have to focus more on the laboratory research as well as the pilot scale up research. There are a lot of challenges which you can abruptly see in the time when we study the drug discovery and development. We see the scenario of drug discovery and development. It is very important that we have to understand a lot of patents are going expires. There are a lot of threats which are around us. If you take the challenges of pandemic, if you take the threats of superbugs, it is very important to think, and being an important part of healthcare system, we have to understand our responsibilities. So this conference, I hope that will be a great success, and the students, the faculty members, my professional colleagues, and all others have taken advantage of this 2 days discussions, because conferences is not going to be very successful until unless we will not take the the basic things from each other. We have to understand all these nitty gritties. So my congratulations to Amity University, their officials, Vice Chancellor, Pro Vice Chancellor, and all of the staffs, the Dean of Pharmacy, all other faculty members for proposing this wonderful uh, content of this conference. And I hope the journey of this research will continue. And my message to through this conference is, if you are doing a science, you have to, I think, this is my own sort of thinking, we all implement it. Triple P concept we have to develop. when we are going for research, we should have a great patience. We should adopt the good process, and we should have the great potential. So this triple P concept should be very necessary to carry out the research. Thank you so much for inviting me and discuss something about this theme. Thank you so much.
Thank you for your words of appreciation and your blessings. They really do mean a lot to us. Thank you for taking time out of your busy schedule. We know your condition for being with us here to bless this event. Thank you. It's now time for self-analysis. Feedback in a conference is crucial because it allows organizers to identify areas of improvement, enhance the attendee experience, build stronger relationship with participants, and ultimately create a more successful future event by understanding what aspects require well and went well, and what needs adjustment based on the direct feedback from the attendees and speakers. Dear delegates, this is to announce that shortly after this event, we are going to share the feedback link, which is need to be filled by you and submitted by today's day end. However, we are also going to request few of our attendees to join us here on stage and share their feelings and suggestions regarding this two-day extended conference. So may I now request one of our participants for the offline oral presentation, Mr. Archit, to join here on stage and share his feeling. He was greatly appreciated by our honorable judges also. So we'd like to hear your experience and your suggestions regarding this two-day event.
Thank you, ma'am. And very thank you to everyone present over here. Thank you to Amity University. And thank you, judges, for the great compliment that I received today. It really boosted my confidence, and it was a great honor to represent myself over here at the oral presentation and to get the chance to share my idea with all of you. And that's all that I would like to say. And thank you so much, ma'am. Thank you.
Can I now request Mohammad Sahil Seki, M.Pharm Integral University, to join us on stage and share his views.
Good afternoon, everyone. We did fine. We will continue in Hindi because it's our national language also. So, मेरा नाम मोहम्मद साहिल सैफी है और मैं इंटीग्रल से आया हूँ और मेरे साथ और भी लोग आए हुए हैं। करीब कल जब रजिस्ट्रेशन की लिस्ट मैंने देखी तो उम्मीद नहीं थी कि इतने लोग आएंगे। करीब करीब 2 पेज इंटीग्रल के बच्चों के नाम और फिर एक एनर्जेटिक मूवमेंट। ये 2 दिन का कॉन्फ्रेंस जो भी रहा, हम लोगों ने एक्सपीरियंस किया। डिफरेंट स्पीकर, डिफरेंट डिसिप्लिन के स्पीकर्स, और बहुत सारी चीज़ें सीखने को मिली। बहुत सारी चीज़ों पे जो ध्यान नहीं जाता है, जैसे बहुत सारे बच्चों ने पोस्टर प्रेजेंटेशन दिए,
ओरल प्रेजेंटेशन दिए। तो कौन सी टेक्नोलॉजी है फार्मास्यूटिकल्स साइंसेस में, जैसे टाइटल भी है कॉन्फ्रेंस का कि रिसेंट ब्रेक थ्रू इन फार्मास्यूटिकल साइंसेस, तो कौन सी टेक्नोलॉजी है, जैसे आपने अभी देखा ऑर्गन ऑन ए चिप, फिर से टेली फार्मेसी, पर्सनलाइज्ड मेडिसिन, स्टेम सेल थेरेपी, तो ये सब चीजें एकदम नेवर है और उभर के आ रही हैं। तो इन सब चीजों से वाबस्ता हुए हम लोग, और इन फ्यूचर हम इस पे रिसर्च करेंगे, रिव्यू भी करेंगे इन सब चीजों को, और अगर मौका मिला तो इस पे कोशिश भी की जाएगी रिसर्च को लेके। कल अनुराधा मैम से मुलाकात हुई, मैंने मैम से कहा कि मैम आपने थर्ड ईयर में फार्माकोलॉजी पढ़ाया है, तो मैम पहचान नहीं पाई। मैम खैर हम लोग तीन ही महीने मैम ने पढ़ाया। खैर मैम आपके साथ सफर छोटा सा रहा, लेकिन आप याद बन गई है उम्र भर के लिए। तो इसके साथ एक सजेशन, जैसे अभी हम लोग और दो तीन लोग हैं, वो अभी इंटरनेशनल कॉन्फ्रेंस ज्वाइन करके आ रहे हैं इंटीग्रल यूनिवर्सिटी में। वो बायो साइंस, बायो इंजीनियरिंग और इंजीनियरिंग डिपार्टमेंट की तरफ से था, और इंटीग्रल स्टार्टअप फाउंडेशन की तरफ से था। तो बेसिकली उस कॉन्फ्रेंस का जो कंक्लूजन निकला, वो कंक्लूजन ये निकला है कि जैसे हम फार्मास्यूटिक्स से, मैं मास्टर कर रहा हूँ तो मैं एग्जांपल दे रहा हूँ फार्मास्यूटिक्स का, कि हमें जो डिग्री लेनी है बैचलर, मास्टर और फिर पीएचडी, तो हमें ऐसे नहीं लेनी कि एक जैसे फार्मास्यूटिक्स में एक हम ड्रग कैरियर उठाएं, एक ड्रग उठाएं, एक डिजीज पकड़े, और उस पे एक ड्रग कैरियर, एक ड्रग और एक डिजीज, और उस पे हमने एक कोई नया सा वर्क कर लिया जस्ट टू सबमिट द थेसिस, और हमने मास्टर ले ली, हमने पीएचडी कर ली। ओवरऑल जो कॉन्फ्रेंस का कंक्लूजन ये था कि फार्मास्यूटिकल फील्ड में हमें एक प्रॉब्लम को ढूंढनी है, हमें एक प्रॉब्लम को ढूंढनी है, उसके ऊपर अपना पीएचडी करना है, फिर उसको पेटेंटेबल कराना है, और फिर उसको कमर्शियलाइज कराना है। हमारे पास नॉलेज के साथ हमारे पास इकोनॉमिकल स्टेटस भी बहुत अच्छा होना चाहिए। बस मैं बस 4 लाइन की शायरी आप लोगों को अर्ज करके और थैंक यू बोलता हूँ कि अल्लामा इकबाल की, कि टू someone recall this line, I am not able to recall. हाँ, थैंक यू। सितारों के आगे जहां और भी है, सितारों के आगे जहां और भी है, अभी इश्क के इम्तिहान और भी हैं। तू शाहीन है परवाज है काम तेरा, तू शाहीन है परवाज है काम तेरा, तेरे सामने आसमां और भी है। थैंक यू सो मच।
Thank you for this lovely diary and your suggestions. We are definitely going to take them into account while having our next events. Since the last 2 days, over 176 attendees have been joining us online, right from 9:30 to 5:30 p.m. So we are also going to have an attendee to share her views regarding this 2-day extended program from online mode. May I request Pragya ma'am to make Anjali Kumari from Himalayan Institute of Pharmacy, Sikkim, to visualize on screen so that she can share her views on this 2-day extended online event. I request Anjali Kumari to make herself visible on screen. Kindly unmute yourself, put on your camera.
Yes, hello.
Yeah, Anjali. Can we just have a few words from you regarding the feedback for this 2-day extended national conference?
Good evening, everyone. I'm Anjali Kumari, student of MPharm, Department of Pharmacology from Himalayan Pharmacy Institute, Sikkim. I had the privilege of attending the two-day national conference on recent breakthrough in pharmaceutical sciences, from bench innovations to laboratory applications and beyond, conducted on 24th and 25th February 2025, organized by MIT University, and it was an enriching experience. The conference was well organized. The conference had featured impressive speakers who delivered insightful presentations on cutting-edge topics in the field of pharmaceutics. The sessions were engaging and informative. Thank you.
Thank you, Anjali, for that brief yet elaborative explanation of yours. Your words of appreciation really mean a lot to us. Thank you.
Thank you, ma'am.
It is now the time for the award ceremony. I request all the dignitaries of states to kindly join us on stage to honor our participants of oral and poster competitions, both at online and offline mode, with cash prizes and certificates. May I request Anuradha ma'am, Tariq sir, ma'am, Ajay sir, please. For online poster presentation, we have Anjali Kumari, the best online poster presentation. Anjali Kumari from Himalayan Institute of Pharmacy. A great round of applause for her. Her certificate will be displayed online. I request Pragya ma'am. Sir, the cash prizes for all those who have been on online mode will be sent within a week. Here we are for the winner of the oral presentation on online mode. We are just presenting the best online presentation at oral presentation at online mode. That is Nabanun Mukhopadhyay. Nabanun Mukhopadhyay, the best oral presentation on online mode. Can we have a great round of applause for N. Mukhopadhyay? May I now request all the dignitaries to please felicitate the first prize of oral presentation offline mode. The first position goes to Mr. Deepak Mishra from AIP AUP Lucknow campus, SRM ICMR. The second prize for oral presentation offline goes to Mr. Archit Akhil from ERA University. Archit Akhil from ERA University. Now it's time to announce the results of offline poster presentation competition. The first position has been bagged by Nandini Sharma, JIIT Noida. Nandini Sharma. Can I request Nandini Sharma to join us online? I mean on the stage. The co-authors are also there. Ravanshi Bhandari and Anjali Sharma. Back with the first position for best offline coding. Thank you, Nandini and Rabansi. Next, we have the second position that goes to Mohammad Sahil Sefil. Mohammad Sahil Sefil, Integral University, for bagging second position for the offline poster presentation competition. I now request Professor Dr. Abhishek Tiwari, sir, Professor, Department of Pharmacochemistry, AIP AUUP Lucknow campus, to offer the official vote of thanks on behalf of Director AIP and the co-organizing committee of RBPS 2025.
सर्वे भवन्तु सुखिनः, सर्वे सन्तु निरामया, सर्वे भद्राणि पश्यन्तु, मा कश्चित् दुःखभाग् भवेत्. May all be happy, may all be free from illness, may all see what is auspicious, and may no one suffer. I feel truly honored to deliver the official vote of thanks for 2 days National Conference on Recent Breakthrough in Pharmaceutical Sciences 2025. As an organizing secretary and my team, Dr. Jishan Fatima, Professor Dr. S.P. Singh, Professor Dr. Varsha Tiwari, Dr. Nimisha, Dr. Lucy Mahapatra. This event would not have been possible without the vision and support of our Honorable Founder President, Dr. Ashok K. Chauhan sir, and Honorable Chairman, Dr. Ashim Chauhan sir. We are deeply grateful to our Honorable Pro Vice Chancellor, AUUP Lucknow campus, Professor Dr. Anil Vasisht sir, for his valuable presence and support despite his busy schedule. A heartfelt thank to Wing Commander Dr. Anil Kumar Tiwari sir, Deputy Pro Vice Chancellor, Director ASET, for always inspiring and guiding us. Your presence is truly motivating. A special thanks to Dr. Kamar Rahman Madam, Distinguished Professor and Dean Research, SNT, Amity University, Uttar Pradesh, Lucknow campus, for your constant inspiration and guidance. Your presence always motivates
us to excel and perform better. A special thanks to Professor Dr. Anuradha Mishra Madam, Director AIP, AUUP Lucknow, for her constant support, guidance, and encouragement, which kept us motivated throughout this journey. A special appreciation to special guest, Professor Dr. Akash Ved, Member Pharmacy Council of India, Guest of Honor, Dr. N. Shahar, Deputy Director, CCRUM, and Chief Guest of Inaugural Session, Dr. Niranjan Babu, Member Pharmacy Council of India. Also thankful Dr. Mahmood from CCRUM. I am thankful to Chief Guest of Valedictory Session, Professor Dr. Neeraj Upmanyu Sir, Member Pharmacy Council of India and Pro Vice Chancellor of Sage University Bhopal. Our speaker for day 1, Professor Dr. Deveshwaran, Professor M.S. Ramaya University, Bangalore, Dr. Sandeep Chaudhary, Dean Naipar Rai Bareli, Dr. Deva Priya, Central University, Punjab. Day 2 speaker, Professor Dr. Ajay Sharma Sir, Dipsar University, New Delhi. Professor Dr. Bibhu Panda, Professor Jamia Hamdard University, New Delhi, Dr. Deepak Chitkara, BITS Plani have truly enriched our knowledge. Thank you for sharing your expertise. We have received 496 outstanding registrations across the country and 200 abstracts for oral and poster presentations. We received registrations from Madhya Pradesh, Punjab, Uttarakhand, Haryana, Uttar Pradesh, Sikkim, and Odisha. We extend our gratitude to Dr. Grain Gautam, Director, Shri Ram College of Pharmacy, Muzaffarnagar, Professor Dr. Anurag Mishra, Director, Shri Krishna College of Pharmacy, Sitapur, Professor Dr. Avinash Tripathi, Director, RGS College of Pharmacy, Lucknow, and Professor Dr. Dharmendra Kumar, Dean, Gopal Narayan University, Bihar, for evaluating the posters. A big thank you to Professor Dr. R. Maitri, Chaityanand University, Tamil Nadu, Professor Dr. Manish Kumar, ISF College of Pharmacy, Punjab, Professor Dr. Tariq Mahmood, Integral University, Lucknow, and Dr. Arya Lakshmi Mary Shetty from Dipsar University, New Delhi, for evaluating oral presentations. I am grateful to our organizing team and all the faculty members of AIP for their hard work and dedication in making this event a grand success. Special thanks to Director IT Vikramji Namanji from Lucknow campus, Arvindji from Noida, Director Administration for Hospitality, Director PR and Communication, and Ammaniji for Media, Director Procurement, Director Security, as well as our office staff and student volunteers of AIP. I especially want to thank you, Nirali Prakashan, for sponsorship. A special mention to our Sahuji for capturing these beautiful moments, and ESCO department. I sincerely appreciate everyone who contributed directly or indirectly for the grand success of this conference. If I missed mentioning anyone, please know that your support is truly valued. Lastly, a big thank you to all the participants who joined offline and all the attendees, those who joined us online on the Zoom platform for your active involvement, making this event truly memorable. I deeply appreciate your support. Thank you. Thank you all. Thank you once again.
Thank you, sir. I request everyone to stand in respect to our national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंधु गुजरात मराठा द्राविड़ उत्कल गंगा। विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा। तब शुभ नामे जागे तब शुभ आशीष मांगे। गाहे तब जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
Thank you everyone for making this two-day extended program a huge success. I would now request all the faculties of AIB and AIB and HODs and HI from other departments to and along with the dignitaries over here to kindly join us on stage. At the same time, I would also request, ma'am, please. I also request the winners of all the competitions to kindly join us on stage for a group photograph. Our sponsor students, some of our sponsor students, Divyans, Ojas, and Anurag. I would also request you to kindly join us on stage. Divyans, Ojas, and Anurag to kindly join us on stage. Divyans. ठीक है. Do we have Divyans, Ojas? Dear delegates, you can now leave for the high tea that has been arranged for you in the back side of the auditorium. And I thank you all for joining us for this two-day extended program.
Certificate they can collect it from the registration.
You can also collect your certificates from the registration desk. After, may I have your attention, please? After having your high tea, you can collect your certificates from the registration desk for those who are outside MIT University. I repeat, after your High tea, you can collect your certificates for all those who are outside MIT University. Students from MIT University will be for you, we are going to distribute the certificates in your classes. Thank you for your kind cooperation. You can now leave for the high tea.